right? And so I show a very famous plot. I'm sure you've seen this many times. So I'm plotting, this is a plot of the viscosity on a log scale as a function of the molecular weight, also on a log scale, for a very wide variety of polymers with very, very different chemistries, right? And what we see here is this behavior which is common across all these polymers, right? And this really arises because of the fact that all these polymers are flexible connected objects, right? So in a sense, the chemistry plays less of a role in terms of determining these qualitative aspects. Of course, the chemistry is extremely important in determining, in deciding the actual values of the viscosity, but in terms of this kind of qualitative behavior, you see this universality that is really just a function of the connectedness of these materials, right? And so the question that we started exploring then is what happens if I take a bunch of entities and I connect them up and I make chains out of them? Do they see different kinds of properties? So in a sense, to look at a colloidal analog of a polymer, but that's a very tall order, so I'm just looking at these as connected objects and trying to ask the question, what happens when you connect these, right? So these, the objective is to eventually look at things that are polymeric analogs, but really this talk is about connected objects. Now, the idea of using colloids as analogs of molecular systems is not new. It's not something that we started. This is something that people have been doing for the last few decades. What are the advantages, right? So we saw this very beautiful talk by Sanjay where he used synchrotron scattering and he's done a lot of electron microscopy to visualize these polymers. These are very challenging techniques, right? They are very hard techniques to use. They're high vacuum techniques very often, and you then have limitations as to the kind of systems you can examine using these. The advantage using these simple colloidal systems is that these are materials that you can observe using simple microscopy, right? They're sufficiently small. If I take a 1 micron colloid, this is an object that's sufficiently small that these are still Brownian, right? So they still exhibit Brownian motion, and yet they're sufficiently large and therefore slow that you can use very simple microscopic techniques to observe these, right? So that was really the idea. So for example, for a 1 micron colloid, in water at room temperature, this would have a diffusivity of the order of 0.1, 0.2 micron squared per second, right? So very easy to observe using simple optical microscopy. So this is something that people have done a lot. So for example, the group at Harvard and the group at, in the Netherlands started looking at colloids to examine basic concepts like the classical nucleation theory for crystallization. The group of Rajesh Ganapati at Bangalore has been doing some very nice work to look at crystals and then looking at amorphous interfaces between these crystals and looking at the glassy dynamics in these regions. There's also been some very nice work again from the Dutch group looking at things like capillary waves, and this is just a small subset, but the idea is to try and convince you that there is a possibility that at least some molecular phenomena, phenomena exhibited by molecular systems can be replicated in these large colloidal systems, with the exception that these are really easy to do in terms of experiments. So we decided to take these colloids and line them up, and one of the strategies we employed is to take these colloids and line them up in an AC field, right? So here's a dielectric colloid, so these are either silica or polystyrene, turns out it doesn't really matter. And you put them between IT-coated glass slides and you put a field across them. And if you put an AC field at a high enough frequency, you have no liquid motion, but you induce a dipolar interaction between these colloids, and as a consequence, the colloids line up in the field, right? So this is something that is a very beautiful phase diagram from the group of Van Bladeren that was published in 2003. The idea is we take a part of that phase diagram and implement this regime where you have these chains that line up in the presence of a field. Now, people have done this in the past. Alice Gass group and Eric First was a PhD student with her, and also Van Bladeren, more recently in 2012, have taken polystyrene colloids. They line them up in the field, and then they heat them to go above the glass transition, so they sinter them, right? So you get these connected objects, but the connectivity is really through sintering at these boundaries. Rather than that, what we decided to do is to line them up, but each of these colloids is coated with a polymeric material, so either a microgel or other kinds of polymeric materials, but these are cross-linkable polymers. And so you essentially coat these colloids, line them up in the field, and while you have the field on, you cross-link the shell around them. So effectively what you're creating is this molecular cross-linked shell that links these particles together, and that gives us a lot more flexibility, said in multiple ways, of connecting these colloids as compared to the previous way of sintering them together. It allows you to get polymeric objects. So now I'm going to call this object a polymer because it's a connected object, right? So these connected objects can be flexibly linked together. And if you control, for example, how long you allow the cross-linking to proceed, here is, these are optical microscopy images. They look like simulations, but they're not. They're really optical microscopy images of flexible chains, sort of semi-flexible chains and fairly rigid chains comprising several tens of these colloidal objects, right? So if you look at this, I'm sorry, I don't think it projects very well on the screen, but you can see this, essentially something that looks like a molecular dynamics simulation, coarse-grained molecular dynamics simulation of a polymer chain that's coiling and uncoiling entirely due to thermal fluctuations in these systems. So really, you have then the analog of these connected chains where by controlling the cross-linking time, you can control the degree of rigidity or flexibility in the system. So of course, we can quantify this. What we have here is a plot of the center to center distance between these chains. Remember, these are micron-sized colloids. So all I need is a cheap, 5-10 lakh optical microscope to look at these systems. So you can look at center to center distance, and you look at the distribution, again due to thermal fluctuations in these systems. And what you can see is that as you vary the cross-linking time, you can vary the rigidity in the system, so you can get larger or smaller fluctuations in the center to center distance. From this, you can pull out then a spring constant, right? What you think of in a bead spring model, this is the spring constant that we can pull out from our experiments. Similarly, what we can do is to look at a dihedral angle between three colloids that are linked together, and also then pull out the chain binding rigidity in these systems. Again, this really comes from the shape of the distribution of the dihedral angle as a function of, it's a distribution function of these dihedral angles. So we can make systems which have different levels of flexibility, and you can characterize these. And so just to keep things simple, to have a one number that characterizes this, we define something called a flexibility parameter. So we take a chain and we look at end-to-end fluctuations in this chain. We are plotting here a normalized end-to-end fluctuation in this chain as a function of time. And you can see that chains that are rigid have very small fluctuations. So the root mean square fluctuations are then characterized as a flexibility parameter. So larger the fluctuations, more flexible the chains, larger is the flexibility parameter. Right? So it's a very simple way of keeping track of what the flexibility parameter is in this system. So just to go back, notice that as the flexibility parameter increases, this increases in this direction, you see a decrease in the spring stiffness or in the bending rigidity in these systems, as you would expect. So let me tell you the first story. The first story is where we take these colloidal necklaces or colloidal chains, and you ask the question, what happens when you go from a good solvent to a bad solvent condition? It's a canonical problem in polymer physics. This is the coil globule transition problem. And what we're looking here is a system where this green object is a 1 micron colloidal entity. So this is the part that we can see. And then on this, we absorb poly N isopropyl acrylamide microgels, much, much smaller in size than this. So these are then we are all familiar with polynipam. This is an LCST polymer. And what that means is you heat it up to moderate temperatures, about 35 Celsius, you get a transition from a hydrophilic polymer to a hydrophobic collapse regime. So if you take these monomeric systems, so that's light scattering, looking at diameter of these microgel particles as a function of temperature, and you see that there is an LCST as you would imagine. So the interesting thing then is if I take these, I'm now going to call this a monomer. If I take these monomeric colloidal objects, and as I increase the temperature, you can see that there is an increase in the aggregation, and this is reversible, right? So if you cool it back, it disaggregates and goes back just driven by thermal Brownian motions in these systems, right? So what this allows us to do is to create thermoresponsive systems. So we took these colloidal necklaces and we started heating these. Again, the temperatures we're talking about are modest. You're going up from 25 to about 45 Celsius. Very easy experiments to do in the lab. And we plot here the end-to-end size of the chain as a function of temperature. And for chains with different degrees of flexibility, This goes from 10 to the minus 5 to 10 to the minus 3. So you're going from more rigid to less rigid systems. You see that the more rigid systems show a small decrease in the size, while the less rigid systems show a much larger decrease in the size, as you'd expect for a coil globule transition in these colloidal systems. And so really what's happening is that the monomer-monomer, so monomer here is a colloid, interactions are more favorable than the monomer-solvent interactions as you increase temperature, leading to collapse of these chains. Right? So essentially, we're looking at the colloidal necklace now in a batch solvent. So we can plot this in a slightly different way to make it easier to visualize. So I'm plotting here the monomer, the local density, and so this is the number of monomers divided by the end-to-end distance cubed. So this is a per micron cubed kind of a unit. And as a function of temperature, here are the more rigid chains, here are the more flexible chains, and you can see, apologies, I think I've switched the color scheme around. Oh, no, sorry. So the more rigid chains to the more flexible chains, you see that there's a much larger increase in density locally of the flexible chains as compared to the rigid chains, right? So this is all fine. There's nothing particularly unusual here, but it tells us that we can go back and start looking at things like the coil globule transition using very simple experiments. Here's the thing that we found very unusual, right? So we are talking about chains that are at an intermediate level of rigidity, so not very rigid, not very less rigid. And as you heat this, there's a little bit of convection because of the temperature change. This is a dynamical experiment, and when you follow this chain, and this is the temperature, once you start going above the LCST temperature, what you can see is that this chain starts organizing into a helical structure, which is very unusual. We really did not anticipate seeing this at all. These helices are, so this is a 2D imaging, so this is hard to tell. What we can do is we can plot the XY coordinate, and on the Y axis, since the helices go in and out of the plane, we can use the Z axis as the intensity that we see, and you can clearly see that if you take these kinds of chains, you can clearly see the formation of a helical structure in these systems. So really we see the formation of very beautiful helices that form. There is no preferred handedness. We don't put in any preference. There is no preferred handedness. You can form left or right-handed helices with equal felicity. These helices form and unfold, so they fold, unfold, refold dynamically over time. So, so it's sort of a phase diagram that you see for this behavior is that, so you're going from low temperature, which is an expanded state, to a collapsed state. If you have chains that are less rigid, I'm not going to call this flexible because these are not truly flexible chains. If you have less rigid chains, you see a large change in the shape, and so you see a collapse in the end-to-end distance. At very high rigidity, so very, very rigid chains, you see a small decrease in size but no change in shape. The most interesting phenomena, in our opinion, is in this intermediate regime where you go from chains that are stretched out to chains that fold to form helical structures in these systems. So if you think about this, the helices are special, right? So people have looked at helices in biology for a long time. This is a very famous example from that talked about the DNA double helical structure. But also in polymers that we know and love. For example, this is an article that I was very happy to get Dr. Sivaram to write when I was editing an issue in Resonance, where he talked about the origins of stereoregular polymers. Now, polypropylene is something that, isotactic polypropylene is something which is very dear to Dr. Sivaram. It's one of the first polymers he dealt with on large scale when he moved to IPCL. Now, again, in these systems, there are directional interactions, right? There's either directional hydrogen bonding interactions in these systems or directionality imposed by the side chain in the case of polypropylene. Now, this is not the case in our system, right? In our system, we don't put in helicity at all. There is no reason to put in any helicity in these systems. There's no directional interactions. There's one example in the literature where they talked about simulations of essentially cylindrical rope-like structures where they put in attractive interactions using what are called depletants or depletion interactions, where they showed the formation of these helical structures. This has not been done in experiments, but really in the case of bead spring kind of structures that we've made, this is really the first experimental demonstration, and to the best of my knowledge, there hasn't been any theory that talks about the formation of helices in the structure. So question is, what really drives the formation of these helices? So we went to our friend across the street, Apratim, who was at ISER Pune, And he ran some really simple simulations of this, looking at very model systems. And to just cut a long story short, what we are plotting here is an order parameter that tells us about the formation of helicity in these systems as a function of the bending rigidity. And what we see is that when chains are very flexible, there is no so when there is a collapse transition, when you go to a bad solvent state, these chains simply collapse and really there's no formation of a helical structure. Similarly, in systems that are very rigid, there is no formation of a helical structure, but over an intermediate range of rigidity, you have collapse that really drives, in these systems, remember these are bead spring polymers, and so there is the excluded volume of the bead in itself, and so in these systems, as these things collapse, there's kind of a strange buckling transition that forces the formation of helicity in these systems, right? And that's really the main story from these simulations. So I promised you 2 stories, so very quickly let me move to the next story, which is to go away from Brownian equilibrium systems to start looking at something that's very interesting in biology, which is the idea of active chains. So these are systems where you have energy that flows in and out of the system. Typically, when you think about an active chain, right, so these are things like, for example, self-propelled objects. So for example, if I have bacteria, you know that if you put bacteria and you observe them under a microscope, you see them being moved in a particular direction and they flip and move in another direction. So these are active systems where they harness, they take in energy either by running chemical reactions or by other means from the environment, and then they use that to perform these kinds of actions. So in all these systems, there is a need to break symmetry, right? And that makes it challenging because synthetically, if you want to make these systems, you have to make systems with some kind of broken symmetry. Janus particles, for example, right? So you take a particle, you coat one side, and you make this, an object which has no symmetry in this spherical system. Now that's a little difficult to make, and it's certainly difficult to scale up to make in large quantities. And so that's when we got really inspired by some very, very interesting work that was done by the Chennai group, where they said that if I have a bead spring object, then Brownian fluctuations in flexible or semi-flexible systems lead to continuous breaking of symmetry. So if I have this chain, right, and you have Brownian fluctuations, you no longer have this spherically symmetric object, right? And so what they did is then they modeled this using the normal hydrodynamic interactions for, so if I write down the equation of motion for the system, and then they put in by hand a term which came through these active processes. This term is driven by this by the symmetry breaking in the system. So this is something that we felt we could reproduce. We used a slightly different technique to make the chains here. We used a technique that we developed called ice templating. So you take these colloidal objects, in this case 1 micron objects. You put in a polymer that can be cross-linked, and then you freeze this dispersion. This is in water, so you freeze this dispersion, and you can get the formation, actually, strangely, never branch systems, but a lot of linear objects, right? About 10% of this yields as linear. So I won't go into very simple hydrodynamic simulations that we were able to use to, Brownian simulations, there's no hydrodynamics in this, but that we were able to use to mimic quantitatively the particle size distribution of these systems. So what we'd shown in a separate piece of work is that when you do this, then you get these polymer chains that assemble around these colloids. These are then cross-linked, and you can get these essentially bead spring type systems. And so we made these chains, and you can make these chains of different sizes, like we've shown before, and we then measured center of mass diffusion for these chains. Again, you take this chain, put it under the microscope. It's very easy to figure out the center of mass, and then you can look at the way in which the center of mass moves with time. And from that, you can pull out this mean square, the mean square center of mass diffusion as a function of time. This is linear, and the slope of this gives you the diffusivity. Okay? So you can then plot the diffusivity as a function of chain length. And what you see for these chains, remember, these are Brownian. These are equilibrium systems, that the diffusivity really depends only on the chain length and not on the flexibility. So for a certain chain length, we look at different flexibility, look at another chain, we can look at another chain length, look at different flexibility. It's not a function of the flexibility of the system. And so you can then plot the diffusivity as a function of the number of particles, and you see a decay that looks a lot like what Zim predicted. that's unusual because you don't think that the ZIM system works for small n systems, but turns out that ZIM's original model was for any chain with Gaussian fluctuations, and so you see the ZIM kind of decay in these systems. So far, so good. So we take these chains and we walk to Prasad's lab, Prasad somewhere here, we got him to give us platinum nanoparticles, absorb them on these chains, and then the platinum nanoparticles are catalysts, and so we put it in the presence of hydrogen peroxide, he decomposes the peroxide to form water and oxygen, and we do this at dilute concentrations, there's not a lot of oxygen that's bubbling out of the system, and then we can start looking at the behavior of these systems. So remember then, Ronojoy and Sunil's argument tells us that fluctuations in the system is what breaks symmetry. So if I have a single colloid, I should not be able to break symmetry. So whether or not I have platinum on the system, I should get the exact same Brownian diffusivity. So that's just a check, and we ran that check for monomers. So this is single beads. Whether you have platinum and you run it in the presence of peroxide or just water, you get the exact same diffusivity, which is as you'd expect, right? Everything is nice and symmetric. You don't expect to see any kind of active processes in the system. The interesting thing is when you start looking at active colloidal necklaces, and what you can see then is that, so here is passive chains, and I already showed you that the Brownian chains, the diffusivity is not a function of flexibility or chain length, but as you go to active chains, and so what we are seeing here is an increase in the flexibility parameter. With an increase in the flexibility parameter, you see a much enhanced diffusivity of these chains, right? The diffusivity of these chains goes up by of the order of 60%. Remember that the diffusivity is proportional to the temperature in an equilibrium sense, and so this is effectively like chains that are at some ridiculous, like 500 Celsius or something like that, right? So this is fairly significant in terms of the increase in diffusivity. This is not, this is really something that is not an experimental artifact, right? And so again, we ran simulations, and so out of time, just to let you know that Ronojoy and Sunil's simulations more or less match what we see in our experimental systems. So I just want to leave you with this thought. So it's sort of an unusual talk for this meeting, but I wanted to give you this idea that you can get these very easy tunable model systems. We looked at systems where we did active chains, we looked at systems where we did thermoresponsive chains, and so this then enables us to do very simple tabletop experiments that give you very interesting physics of these kinds of connected objects. And to let you know that we're currently working on sheet-like objects, we're very interested in dynamics of these and what happens when you have holes in these sheets and so on. So with that, I'll stop. I think I'm on time. Thank you.
Thank you, Guru, for being absolutely on time, or rather before time, and giving a wonderful presentation. The floor is now open for questions.
Hey, Guru, lovely talk here. I really enjoyed it. I was just curious to know your number of beads that you've linked are still fairly small on when you think about statistical properties of polymer chains, right? So I was wondering whether, you know, I saw your last portion where you varied the n, but that's still not very long, right?
Yeah.
So it's quite interesting that even at that small lengths, they actually bring out a lot of the expected physics. So do you have any comment on that? I'm just a bit,
Yeah. So thank you for bringing that up, RK. It's something I didn't have time to go into. The second part of the talk where I talked about active chains, right? We deliberately restricted it to small n because once you go to very large n, then you eventually lose symmetry in these systems. So you have large scale flows which are then back to symmetric, and you don't get this kind of an effect. So this effect is something which is very visible when you have a limited number of beads. In the first part of the talk, when I'm talking about thermoresponsive chains, I have to have a minimum number of chains so I can form a helix. So those were chains where you had, of the order of 50, 80 kind of beads linked together. So those are much larger. If you think of each bead as really a Kuhn segment, then this is really getting into the regime of, we're really thinking of short polymer chains, but polymers and not oligomers. So to address your question, so we could, in principle, do these to longer chains. So for example, if I put them in a field, depending on how big, what the spacing between my electrodes is, I can get them to be even longer than that. Right? So we can do this, but so when we saw the ZIM kind of dynamics, we were astounded because we wouldn't have expected to see that. except when you go back and look at the original papers, then it's kind of clear that it does work, so even for these systems.
Guru, thank you for that wonderful talk. So you mentioned about the temperature-dependent, helicity formation, which is very interesting, but you don't have any specificity.
Yeah.
That's what you said. So I'm wondering whether is it possible to bring in some sort of a preferential helicity, either P or M, by bringing a point chirality to your colloid?
Yes, absolutely. And one of the wonderful things about this field is that we leverage the advances in the technology that exists for making these kinds of colloidal systems and just microscopy, right? So, but this is something which we don't do. And so, if you have, for example, materials that you have which have chirality, I mean, it could simply be magnetic. So it doesn't have to be sort of geometric, but it could be some kind of a magnetic beads in the system. We'd love to be able to play with those. Exactly. But we don't know how to do that. So if you can, because if this is, so we work with colloids that we just buy off the shelf. But if there are systems that you think can be engineered to do those, I know this exists, right? But we don't have the synthetic expertise to do that. But we'd love to be able to talk to people who can. Great. Thanks. Thanks, Ajay. True. In this part, yes. But if I can now take that ahead, and I think that's what Ajay is saying, and get specifically L or R, that would be even more interesting.
Guru, wonderful talk.
Hi, Prasad.
So the first part, where you have a bead and then a microgel or whatever it is it important? It's like a core shell kind of a system.
Yes.
Right? And the core is a little rigid than the shell which is flexible.
Yes.
Is it necessary or it's not really necessary?
So I have a core shell structure. So the construct that we have is basically the shell is bonding. Right? So the shell is very, very small. In the second case, for example, I simply have polyethylene chains. So it's essentially nanometers in size. In the other case, since you have microgels, it's a little larger, but it's still on the order of tens of nanometers in size. The shell provides me the bonding, right? So essentially, it's an equivalent of how I induce bonding in these systems. Right? So an analog of molecular bonding. So yes, in my system, it is required because I don't have another way of bonding them. The other point is the shells, the core is not covalently coupled to the shells. If I did, then I wouldn't be able to get large deformations, right? So they are adsorbed, and the whole thing is, it's sort of like a mesh, right? So I have a cross-linked mesh that encapsulates the core.
Yeah, thank you.
Yeah, I thought I will take the advantage of being the chair to ask you a question, but for the interest of time, I'm going to ask you later on. And we will move on to the next part of this session, which is a panel discussion, which will be moderated by Dr. Premnath, who is the director of Venture Center. And I'll hand over the mic to Premnath to introduce the panelists in the...
Tell him to keep it on, please. Javed, keep it open, please. That's okay. Will it really hit their face? But I just wanted them to speak about their own things. So, yeah. Is it okay, Rani? Yeah, start it, please. Okay, let's get started. Good morning, everybody. My name is Premnath. The session is on innovation and entrepreneurship, and I thought this will give many of the students and scientists a perspective on some aspects of innovation and entrepreneurship, and you get to hear from entrepreneurs themselves, many of them working on systems which have polymeric materials involved in them as well. So let me get started. Can I get this on, please? Yeah. While I'm doing this, I'll request the panelists to please join here. There might be a little bit of an issue with the slides, but I hope all of you can join in here. So I'll request Sachin Dubey over there, Parul Ganju, Tanuj, please come, sit here. Yeah. I see Anuya there, and Jayant Khandare, please. Yeah. Yeah, that's fine. Can I just next? Yeah, okay. So the plan today is I'm going to take a couple of minutes, few minutes to just give you some opening remarks and also talk a little bit about Dr. Sivram's contributions in this field, at least the ones I have been involved with. Following that, a few comments about startups and entrepreneurship, and then please take the seat. I think you should be okay. Yeah, it'll take some time. Can I? Yeah, my plan was to ask each of them to introduce their companies while I put up the slides. So please bear with me. We'll move the chairs as we go along as well. Okay. So first of all, I'm trained in polymer science and technology, but my interest is seeing products in the market and in use. As a biomed engineer, I like to see patients actually using products. So that's where I come from. We've been working on several things with Dr. Sivram as well. Besides technology development, we have been looking at innovation management, tech commercialization, and scientific entrepreneurship as well. Just a couple of points. Today, I think Dr. Sivram finishes 75 years. Happy birthday to him. Today is 4th of November. A few comments from my own perspective. There are scientists and then there are technologists among scientists. So to me, Dr. Sivram exemplifies technologists among scientists, people who solve problems with their science, and that is something which is outstanding. Of course, all of you know him as a prolific inventor, but one of the other remarkable things about him has been the technology foresight that he brings and the strategy that he can bring to bear on different areas. And there are many domains where he has tried to flesh out the strategy as well as create the large picture so that many other people can contribute to it. Amazing to see him keep track of information, lots of information from the scientific literature, the patent literature, the trade literature. I don't know too many scientists who track the trade literature, and I think Dr. Sivram is one of them. And we have worked together on some of these. We have been monitoring, for example, we used to monitor technology for polyolefins segments. And of course, he has done for polycarbonates as well. There aren't too many people who have hands-on experience with IP, technology transfer, collaborative projects, innovation management, and so on. And of course, he's engaged with several entrepreneurs. Today, I'll talk about first-generation entrepreneurs like those who you're seeing here. But I must mention that he works with a lot of family businesses as well. A lot of entrepreneurs who have built businesses over generations and have contributed in very large ways. And for all of us, he's a teacher and mentor. I've interacted with him personally on several programs. Of course, with the GE-NCL programs or Reliance NCL programs, which have been very large, had many dimensions to it. In 2007, we created the NCL Innovations Research Center to look at various delivery vehicles to market and also innovation management practices, upgrading them, taking them to a new level, looking at assessing technology, building policies, processes, both at the NCL and CSIR level. And we have done many of those things. The ones in green, we had some success. The ones in red, we failed. But I consider them as brave failures. For example, the CSIR Tech was the first effort to do a for-profit tech commercialization activity in India. And I think that is something where we tried to imagine the possibilities, make it happen. Of course, sometimes when you try things, sometimes ahead of their times, it does fail as well. And of course, Venture Center, which Dr. Sivram was the director when we got started. He was responsible for aligning all the stakeholders in the very beginning and getting things going. And of course, the intellectual property group at NCL, which is also a very small group, but manages about 30% of CSIR's entire portfolio of patents, right? There have been a lot of startups and spin-offs from NCL, and many of these originated because of several efforts from NCL to create mechanisms to promote spin-offs and also scientist-led entrepreneurship. So there's a program in the CSIR called the Scientist Entrepreneurship Scheme and several other schemes. I can tell you that a lot of drafting happened here in NCL for many of these schemes and was extrapolated to many other organizations in the country as well. These are some examples of spin-offs and startups from NCL. You hear some of them. I have a few words about Venture Center, which I want to tell you. This started 15 years ago. Dr. Sivram was the director of NCL at that time. We wanted to build a platform for scientists, entrepreneurs, and scientific entrepreneurship to flourish. It's 15 years now. We have demonstrated that it's possible. There were a lot of naysayers in the beginning, and I'm glad that Dr. Sivram was supporting and backing all of us when we were trying this out. And of course, I want to tell you that because he originated this activity as well. What you see there is which was an abandoned campus in 2006, which was thankfully provided for building some of these innovation and entrepreneurship activities identified by Dr. Sivram to take it up. ISER Pune was also born in this campus, the NCL Innovation Park. Today it houses the Venture Center. We have won several awards in the last several years, including the National Award, Technology Business Incubation, a Nation Award for Incubator of the Year, National Entrepreneurship Award in 2019, and this year there was a National Award for Nurturing IP for 21-22 as well in startups. And of course, we are also rated as the #1 bioincubator in the country today. There are several companies which have done first in the world, first in India kind of technologies, and here are some of them which you might have heard of in the news as well. The first citywide demonstration of organic waste conversion to CNG, run some of our bio-CNG driven buses in Pune city using municipal waste from Pune itself. CDSU approved circulating tumor cell diagnostic solutions. We have Jayant here. He's an alumnus of NCL. He's going to talk about it today as well. This is the first one from India to get approved and in the market and in use today. Several others, dual-powered hand crank defibrillators, spinal implants, process modeling, biosurfactants, drones, single-use bioreactors for vaccines, which of course was very important during COVID. And of course you heard about my lab as well, which is today the second largest diagnostics company in India, and launched the first RT-PCR, approved RT-PCR kit out of India, the first rapid test, the first self-test, and today the 45-minute RT-PCR at airports, okay? Plus many other things, including TB and so on, that is coming up as well. There have been 700 odd entrepreneurs we have worked with, a lot of activities involving spin-offs as well, around 11 spin-offs companies, and some very specialized facilities. A lot of women entrepreneurs, about 30% of our founders are women. In the last several years, 7 years, these companies have raised about 600 crores in risk capital and about revenues, clocked revenues of 1,400 crores in the last 7 years. So there has been impact, and I don't know Dr. Sivram, but you may not have thought that that was possible at that time because there were so many naysayers that we were listening to. A couple of quick sentences about startups, then I'll give the floor to our entrepreneurs as well. Startups are a special class of businesses. All businesses solve problems for customers, and there's a revenue model around it that they build. And eventually they have to be sustainable. But the interesting thing for us as scientists is that many startups actually look ahead into the future. There's foresight in the problems that they solve, there's foresight in the solutions that they bring to the table. And that aligns very well with what we do as science, because we are in the business of exploring new knowledge, getting to the boundary and the front line of science, and using that to solve problems. Of course, when they succeed, they grow very rapidly, but that whole process very often involves these early years where you don't know whether it will succeed or not, right? And there's a lot of uncertainty around it because it's foresight driven. You're looking into the future and you don't know that the future is going to pan out. But it is very different from a typical small or micro enterprise which looks at opportunities of today. We are talking about opportunities of tomorrow, okay? And looking ahead into the future. And when you look at the future, very often you have to come up with new solutions, and that's where innovation comes in. Innovation is about building new solutions for problems. And very often it originates in new problem definitions and new toolkits that you develop. Some of you might be developing toolkits in polymer science, which will get used in solving some of these problems. And they involve systematic de-risking, and that's why science is involved, and very often rapid scaling. And this rapid scaling is the reason that it attracts risk capital. This is not a low risk, high reward business. This is high risk, high reward, and the kind of funds it attracts is very different than what you would see, say, a bank investing in, or you doing in an FD, and so on and so forth. This is not that kind of money, right? This is looking at opportunities for the future. And this is important for the country because this is what drives the future opportunities for the country, both economic and also in terms of problem solving. Science-based startups are a class under this which focus on innovations which are more technical, and also very often they look at problems which have been defined because of new insights from science. Parul will probably give you an example of that. Their new understanding of how vitiligo progresses has led to new drugs, and she can talk about that as well. And of course, there's systematic scientific studies involved, and the challenges are very different than a typical startup. So over to the panelists now. I'll request, after this slide, I'll request you to speak about your company. Sachin, you might need a mic in your hand. I was hoping, yeah, you have it. Okay, good. Sachin Dubey is the CEO and co-founder of Module Innovations. He is doing some very exciting work in diagnostics. Many years back, he visited NCL and worked with Dr. Jog in the Polymer Science and Engineering Division. And there, his journey into entrepreneurship began. He is a master's, he's done an MTech from the University of Rajasthan Center for Converging Technologies. He's he's one of the people who's, in my opinion, a great communicator of some of his ideas. He's been able to convey to a very large audience of different types the value that he brings to the table as well. And he has been project leader in several projects, and most recently, he's won the prestigious Carbex grant, which is the largest grant program for antimicrobial resistance research in the US. And that has been driving some of the work in his company as well. So Sachin, I would request you to talk through your company. Your slide is up there, and then we can go to the next person, please. Yeah.
Thank you, sir. I actually feel really humbled and overwhelmed sitting this side, because if I date back in 2014, actually I was working as a student intern. So unlike others who had a job experience prior to starting or had some other experience, I was actually a student. So for all the students, I think this could be a relatable story. So with that, the journey of Module Innovations actually began from NCL. Module Innovations is a startup which works in the area of precision diagnostics for infectious disease and also tackling the problem of antimicrobial resistance. Currently, Module focuses on the problem of UTIs, which is a urinary tract infection, and we see this as one of the major drivers of the rise of antimicrobial resistance. I would also want to talk about, how the journey began from the labs at NCL to eventually moving out to Venture Center and then eventually moving out of Venture Center. So back then there used to be a program called as Directors Fund, something like that. And we got that small kitty of fund to actually try out the ideas that we had, which panned out that we were actually able to demonstrate those concepts that were only ideas at that point in time to an institution called as BIRAC. We started pitching to them, of course, with guidance from Premnath sir. We failed in our first attempt. And eventually, the lesson of persistence came in. We pitched to them again. And eventually, after that, there has been no looking back. So in a nutshell, we own our roots to NCL, came as students, and now Module is one of the leading names working in the space of antimicrobial resistance. There are two classes of products that we are making. One are rapid tests, which are very similar to how lateral flow tests look, but eventually they try to actually understand if you have a UTI right at the point of care. And also to top it up, we are building solutions to identify which could be the most effective antibiotic and what could be the minimum inhibitory concentrations in as short as 2 hours. So we're building a test called as AST Sense, which is the world's fastest 2-hour test, which is phenotypic to determine the MIC. Yes, so I think that is all about Module Innovations. It started with 2 people. Now we are a team of almost 14, 15 people outside Venture Center. And still I would call we are a startup and would want to grow further.
Thank you, Sachin. I understand people can't see you, so in future I'll just request others to just stand up for a few minutes. What I'll do is after these slides are done, we can all sit down and discuss, okay? Okay, over to Jayant. Jayant, you're next. Jayant, of course, is an alumnus of NCL. We're very proud of him. He did his PhD here, went on to do postdocs in the US, came back to work in the Indian pharma industry, and then decided to set up his own company. He's also an academic. He's a prolific inventor and publishes a lot. He will, of course, this particular technology he will tell you about, but it's the kind of science that is behind it is outstanding and something which is an example of taking high science to the end point where you can do reliable testing for patients. So over to you, Jayant. Please talk about your company and also about your technology a little.
Let me talk about my journey. I walked in NCL 23 years back.
Yeah, sorry. Here.
So thank you, NCL, who allowed me to walk in this space 23 years back. And, privileged to be with you today. And I owe everything what we achieved for impacting human life in terms of saving or extending cancer patients' life to this space. What we have done is actually we have achieved an incredible task by achieving India's first medical device that has been validated in this country for our cancer patients at very affordable cost, 1/10 of the price that actually this technology is available currently in US. As a first medical device company, we have developed a technology called as an OncoDiscover, which is used for detecting early metastasis because 90% of patients, when they come into the clinic, they are actually with the no treatment possibilities. And I can proudly say that this is only second company in the world who has the regulatory approvals. I also have a second pleasure to say that we are going to set up our lab in Chicago and San Francisco in next 15 to 20 days from now. So I think impacting human life is all probably a primary goal of any researcher or scientist, and I'm very glad that OncoDiscover is part of that mission. So thank you, DBT, NCL, DST, all my colleagues. Most of my research team is some form of actually NCL students and alumnus.
Do you want to talk a little bit, Jain, do you want to talk a little bit about your technology as well? Because this is a scientific audience, they would be curious.
So we have developed a multi-component system which is based on antibody-mediated multiple components like carbon allotropes, dendrimers. Those who work in polymers, they know all this kind of an hyperbranch polymers, including magnetic nanoparticles. But the spacing and the arrangement of this component functionalities has been managed so well that the sensitivity of picking cancer cell from cancer patient's blood from 1 ml is the most Herculean task in a science that actually today says about. Why? Because the abundance of circulating tumor cells that originate from primary tumor and comes into the blood in the form of a circulation are distributed to the distant organs. That's how the metastasis progress. And that's why 90% of the patients who are treated for the primary cancer are ignored for the secondary cancers. And that's the mortality that actually achieved. So while we have actually given head and neck cancer is India's cancer, but we have given to the world that head and neck cancer is no more a local regional disease, but it is a systemic disease because it's India's cancer. Unfortunately, man-made etiological a problem. It's in fact impacting many states 300 times. So the challenge in science of actually picking those rare cells from billion of other blood cells is most important. And that's what I think NCL has taught me a chemistry. I communicated Dr. M.G. Kulkarni today, who was my PhD guide. I learned part of those components of designing materials which has very, very specific component. I am very glad to say that we do test patient's blood sample from India, from Kashmir to, say, Kanyakumari. We have done this now abundantly. We have saved, I will say, thousands of lives. The progression of a disease from primary to the secondary is actually to the circulating tumor cells, and we pick them with the highest efficiency and specificity by our publications and records that actually we publish into ASCO and AACR. So it's a long process. Startups are, they are the aspirations, I think, with a frugality that with the limited resources, what we have to have is actually unlimited dreams. And that's what we have done. And we have been now in multiple joint ventures, millions of dollars of funding. I'm very glad that we started with something and we are into genomics, where I think we are the world's precision oncology company who determines now 600 mutations from cancer patients' blood rather than the tissues. And even this part of a science translation is going to happen in San Francisco very soon.
Sure, thank you, please. Yeah. Well, if some of you are curious, you can look up OncoDiscover as a brand online. And their brand in the US is going to be OneCell DX, which you will also find online very soon. Over to our next panelist. Sorry, I had a slide for you, Jayant. I forgot to turn it on. But I must also point out that Jayant's technology went through extensive testing and validation with a lot of support from people like Tata Memorial Hospital, which is the largest cancer hospital in India, the All India Institute of Medical Sciences as well. And it is, he has a facility in Pune as well, in Wakad area. If I'm sure students you will welcome, right? Okay, so over to Dr. Parul Ganju next. Parul, the light seems to be coming on the face, so you might have to go a little bit to that side. Dr. Parul is also a product of CSIR in a way. She has an association with IGIB. She did her PhD from the National Institute of Immunology in Delhi and was a DST Inspire faculty at CSIR IGIB. And her work, which is pioneering work in the field of vitiligo, which is an autoimmune disease, was the genesis of this company. They are actually a therapeutics company, a rare company in India. There aren't too many therapeutics companies out of India, and they're doing extremely well. She will tell you more about it. Do, of course, talk a little bit about the technology also, but at the same time, also where you are today in this development process. Many of you might be aware there's a lot of action happening in the autoimmune disease space right now. A lot of it is happening outside India, of course, but we are proud to have a company here which is breaking new boundaries. One of the things I must mention is that some of the initial work from IGIB and NCL was involved in getting Parul off the ground, right? Of course, now she has other molecules which she is pursuing as well. So over to you, Parul, please.
Thank you, Dr. Premnath. And yes,
Little
indeed,
closer, please. Yeah.
the work started off from the CSIR Institutes, IGIB and NCL, where I did my PhD from NII, and I worked on understanding skin diseases, skin pigmentation, and that's where the interest in vitiligo developed, which is an autoimmune disease where white patches appear on the skin. The genesis of the idea, the science behind it, came from the understanding that I and my co-founder picked up from the work that we did in academic institutes, but we were very keen on translating that science that we have developed into, as Dr. Jayant said, impacting the actual patient population, and we could only do that if we were to set up a startup company, and that's why the strength and or the to start this Ahamune Biosciences came up. I'm very proud to say that in the last 6 years, we've reached from the discovery stage to now a clinical stage where one of our new chemical entities has reached clinical trial stage, and we've very recently completed a phase 1 clinical trial where that NCE was found to be very, very safe, and we are very, very excited to now move on to the phase 2 trials. Again, this is one of the fastest that any company in the world would have reached this stage, right from discovery to development to now the clinical stage, and that has all been possible because of the hard work of our entire team and the wonderful mentors that we've been very fortunate to have. Just a little bit about the technology. What we have done is we've identified a new pathway for intervention in vitiligo. Now, as Dr. Premnath said, this is an autoimmune disease. Now, the only thing that is available currently, which is prescribed for vitiligo, automatically becomes immunosuppressives. And that's true for most of the autoimmune diseases which are currently known. The problem with autoimmune diseases is that because no target is known to specifically modulate that specific cell-specific, organ-specific pathway, there are a lot of side effects associated with immunosuppressives, which is what we want to change. Now, what we have done is, as I said, we've identified a pathway that causes the trigger of autoimmunity in the skin, leading to loss of melanocytes, ultimately developing pigmentation disorders. With this new chemical entity, we are able to not only stop these triggers of autoimmunity, but also bring back color to these white patches. So they are working in a very holistic manner. And this clearly means that this is a first-in-class mechanism of action, never before tested in the world. And what we've done is we've protected these NCs with the IP assets. That's basically the company's asset. And now this molecule, one of our molecules, has reached clinical stage, completed phase 1, and we are simultaneously developing on other skin diseases to maintain skin homeostasis for diseases like psoriasis and atopic dermatitis as well.
Thank you, Parul. Please. Yeah. Well, again, I forgot to switch to your slides, please. Tanuj, you're next. Tanuj is the co-founder and CEO of Nayam Innovations. They're doing some very interesting polymer science. I'm sure many of you it'll pique your interest. Tanuj has done his engineering physics from IIT Bombay and leads a team which, by the way, includes very eminent chemical engineers, polymer chemists, and so on and so forth. And ophthalmic surgeons with a lot of experience. He's previously worked for an investment banking firm, Credit Suisse, decided that was not his cup of tea, He wanted to be back in technology development, developing technologies. And so he's taken on this very challenging problem of developing the next generation of intraocular lenses, which will hopefully revolutionize how cataract surgery is done in many parts of India and the world as well. So over to you, Tanuj. Can I request you to just come here a little bit while you speak? Yeah.
Thank you, Dr. Premnath. So I can owe the genesis or the founding of NIAM Innovations to Venture Center. So it was through Venture Center I met two of my co-founders, Dr. Suren Ponratnam, who many of you, who was a colleague several years ago, and Professor Julie Confield, who is a professor of chemical engineering at Caltech. So it was through Dr. Premnath.
Yes.
Yeah.
Thank you.
So it was through Dr. Premnath and Dr. Guruswamy, this chance meeting happened, and we decided to set up or incorporate this company. Previously, I had founded another company into chemical engineering process design, and it's a different story how I moved from applied physics to investment banking to chemical engineering and now into polymer science, but that's something for later. Moving on to the technology, cataract is the most performed surgical process in the world. And in cataract, our natural lens opacifies due to age, and our vision deteriorates. The only treatment for cataract is that we should replace our natural lens through an artificial intraocular lens. But intraocular lenses which are currently in the market, they're not personalized or customized to individual patients' optics or neutralize all the errors in the optics of the human vision. So as a result, after cataract surgery, there are many patients are dependent upon spectacles, or sometimes these refractive errors are left uncorrected. And in bottom of the pyramid, if the patient is of the bottom of the pyramid class, then he cannot afford spectacles, and therefore his quality of life deteriorates. Okay. So we have invented a technology platform by which we can make a patient personalized intraocular lens, which is customized to each individual patient. So essentially we have invented a polymer composition which can change shape, whose topography and topology can be changed upon exposure to light. Okay. We have invented the manufacturing methods to cast this material into an intraocular lens. We have also invented the software and algorithms which is used to calculate the personalization required for each patient. And lastly, we have also invented the instrumentation or the optical instrumentation which can be used to precisely shape the intraocular lens. So this technology works basically on diffusion-induced shape change. So on a polymer, in a polymer matrix, there is macromers, low molecular weight macromers, which are free to diffuse inside the matrix. And when we shine light on this composition, then part of the macromers are polymerized, and it causes diffusion-induced it causes diffusion-induced mobility of macromers, which effectively leads to mass transfer, and which changes the shape of the lens. And this process can be controlled very precisely. So we started from an idea. It was just idea on the back of a paper, where I still remember that night where Dr. Julie, Dr. Ponratnam, Dr. Premnath, we were meeting in person and on Zoom. Not Zoom, Zoom wasn't there at that time. I think it was Skype, yeah. So it was that long ago. So we started from an idea, just an idea on the back of the paper. And I think due to our team and the mentors and the terrific support system provided by Venture Center and NCL, we have finished our animal studies and preclinical studies, and we'll begin human clinical trials next year. So thank you very much, Venture Center and NCL, for all the support you have given us.
Thank you. Please. This is Tanuj's facility in the city, which, of course, he has moved out of Venture Center now, and this is his new facility out of in Baner, right? It's not the entire building, it's some part of it. In case all of you are wondering as well. Over to Anuya now. Happy to have Dr. Anuya Nisal out here. She's, of course, principal scientist in the Polymer Science and Engineering at NCL, and also the director and co-founder of Serigen Mediproducts Private Limited. Anuya is trained in polymer science and in chemical engineering, has worked in GE before, and before joining NCL. And then in NCL, she has built a program around silk-based materials, and that was the inspiration for some of the products that she has been building in Serigen Mediproducts as well. There's a dedicated team in the company which is building out the company right now. And over to you, Anuya. Please tell us a little bit more about it. Of course, I must also tell you that Anuya she's been recognized by the Indian National Academy of Engineering by the Young Entrepreneur Award. She's also a winner of several awards, including she was the national winner of the TIE, which is the Indus Entrepreneurs, the world's largest entrepreneurs network amongst women entrepreneurs. And of course, the national award for technology innovation from the Government of India for polymers in public healthcare as well for 2019 and 20. Over to you, Anuya, please.
Thank you, Premnath, and thank you for the opportunity to be a panelist on at the SPSI-MACRO. Yeah, so I'm the...
Yes.
Yeah. Today I'm going to talk about Serigen, which is a company that is based on a technology that was licensed from NCL to the startup. So basic technology is around silk. This is the same material that has been used in textiles for several centuries. Very interestingly, the silk thread can also be used to stitch wounds. So silk-based sutures have been in the market for more than hundreds of years. At NCL, what we have done is we have developed protocols by which you can convert the silk thread into a solution. And this solution can then be leveraged and converted into various three-dimensional shapes like microparticles, blocks, films, pellets, and we have very precise control on the porosity, the mechanical properties, the rate at which the silk will absorb in the body. To just tell you a little bit about the genesis of Serigen. So Dr. Ashish Lele actually had a PhD student, and they did some pioneering work based on silk-based materials way back in 2006. So when I started my PhD, we also focused on looking at some very fundamental basic understanding about the molecular structure of silk, how you can process it, how you can control the conformations around silk, the rheological properties of that material. And while we were discussing between Dr. Premnath, Dr. Ashish Lele and me, it was very clear to us that this is a huge opportunity. So the silk has been used as a medical material for several hundreds of years. India is today the second largest producer of silkworm silk. So we have a very mature silk industry. We employ more than 8.6 million people in silk. And very contrast to this is our healthcare sector, where we are heavily reliant on other countries. We import more than 80% of our medical devices. So we looked at this as an opportunity to actually bridge this gap. So connect between the mature sericulture industry and the healthcare industry. I think it was also very important that while we had this idea, what helped us take it forward was the conducive environment here at NCL. We already had Venture Center, which was set up where you could incubate a startup. There was a scientist entrepreneurship scheme, so there were mechanisms by which a scientist could actually take your idea your idea into a company and take it forward from there. There were also grants from BIRAC, which were at a very early stage in the the journey of the startup. So just like Parul mentioned, we began at an idea stage in 2015. And for a medical product, it's a long journey. So you ought to have a grant kind of this, because raising angel investment or capital at an early stage for a medical product is very difficult. So that is how Serigen started. We today have a product portfolio that includes 3 products, both for soft tissue regeneration, hard tissue regeneration, and also in wound care. So our product portfolio caters to orthopedics, breast cancer, and advanced wound care today. Just tell you a little more about Serios. Basically, it is a material that can be used to fill cavities that are formed in the bone either due to an accident or due to cancer or due to trauma. Very happy to share that we are the world's first company to successfully complete a clinical trial by using silk for bone repair. We've seen very good data in patients and no complaints from surgeons. So we are excited to take it forward into the next stage of clinical trial. We've also incubated at Venture Center so far, and in 2020, we have actually set up our own facility, which is now ISO 13485 certified, and also has government approvals where we have launched our first product, Seriderm, in the market earlier this year.
Thank you. Yeah, I think you can keep that. Okay. What I'm going to do is take on a few questions, but I'll start with mine. In the meantime, if people want to ask questions, they can raise their hand as well. Can I request each of you to please come here? I'm sorry that the light was coming on the face, so, okay. So, first of all, I just want to ask each of you to comment on one topic which comes across many scientists and students' mind, right? I want to understand what was your motivation for getting into entrepreneurship, right? And why did you decide that this was the topic you want to bet upon? Yeah? So I'll start with Parul first.
It was a very simple motivation. I just wanted to impact, just wanted to help people because while I was working on vitiligo, I actually got to interact with a lot of patients who were suffering from this disease, and that's when I realized that there was so much of a need for something. And if as scientists we know so much about the disease, if we are not able to help them, then who else will? That was the simplest motivation that we had, to start the company.
But Parul, you could have been an academic somewhere.
No, I also realized translating science from academia is not easy because I studied the entire process of how a new drug gets developed. There's so much of interdisciplinary science that is required that as an isolated academician, I can't do it, A. Second, I think I can say that there are certain very slow processes, because of course, there are government regulations involved. The time that is required for you to translate your idea or take decisions has to be absolutely quick, which I did not see that in academia. That was my experience. I might be wrong. And that's why, it was a very organic decision for us to start a company and take the idea forward into, of course, a risky decision, but, that's something which one has to take. Otherwise, who else will?
Sure. Of course, this audience has a lot of academics around, so
that's why I said it's my experience. I might be wrong, so don't take it.
Over to you, Sachin. Sachin, what was your motivation coming from as a young student getting into it? Is I think very often a very big challenge as well for many youngsters. Maybe you should tell your perspective.
So, of course, while I was doing my internship here, a lot of my friends had, chosen the path to do a PhD. And I was also trying to, see that can I get a PhD. I got few admissions in the US as well. But at that time, there used to be a competition which is called as Able Best, which is Biotech Entrepreneurship Students Team. We had been a part of that. We won, we were the second winners pan-India. And that was one point I thought that, okay, this is one area which, of course, will have a lot of impact by the science that we are trying to do. Additionally, I would say I come from Rajasthan, which is known for entrepreneurship. Back in my college also, I had a startup which was in the space of solar energy. So during my time at NCL, I talked with Premnath sir that this is what we have. Can we, translate that into a company? So that was one thought, of course. And parallelly, to understand the gravity of the problem that we were trying to solve of UTI, my sister-in-law she was pregnant, she got a UTI infection. And even in those scenarios, we did not have a faster diagnosis. So, an empirical prescription was done, she was taking antibiotics, and it came out that she was actually not having a UTI. So, we pondered deeply, understood how, grave the problem of UTI is, and how demonic it will become that if you keep continuing, taking antibiotics. So, that was the time I decided, okay, PhD is something I would not want to do, no offenses, but I would take this path. Of course, it came with challenges. While people who took up great jobs were doing PhDs, were earning handsome monies, and we were actually surviving on the NCL RF internship money. But then, yes, it all paid off. When we see that, if you go to a doctor and in as short as 15 minutes, you can confirm that this is a UTI or not, antibiotics are prevented. So we really feel happy about that. Yes, I think that was the prime motivation.
In fact, in August, September, ICMR has put out recent data showing how rapidly the susceptibility to antibiotics is decreasing in India right now. And it's actually an alarming situation, which I think sometimes I worry about. Are we sort of living in a fool's cocoon world where we are just, we are unaware of what is waiting to happen. So very happy that you're working on it. And also, Carbex is an extremely competitive program, and you're probably the only diagnostic company out of Asia which was supported under the Carbex as well. So that's fantastic work over there. Tanuj, over to you next. Tanuj, of course, was at IIT Bombay. And in IIT Bombay right now, it's very fashionable to go into finance and consulting kind of jobs. He also landed up as an investment banker and then turned course. So maybe you should tell us what was your motivation to become an entrepreneur, and also why did you think that this topic was worth your efforts and time specifically?
So the motivation, there is a history behind that. Now at IIT Bombay, I had the privilege of working during my master's thesis, I had the privilege of working with one of my professors who eventually became one of the leading scientist entrepreneurs in the country. I think his company is one of the largest lab-grown diamond producers in the world. So after working with him, I joined an investment bank. And at the investment bank, I was looking left, right, and center at startups, companies which were startups once, doing IPOs, making fantastic products. And I was talking to the founders of those guys, of those companies. And I realized, what am I doing here? Why am I just analyzing these companies? So I decided that, after 2.5 years of working into an investment bank, I think I'm losing out on a lot of things. So I decided to quit and start a chemical engineering process design company with a couple of my friends at IIT BHU. We did indeed develop one of the technologies which was licensed to another startup. But things were not very happy, didn't progress as we anticipated. And after that, I started, I came to Venture Center, and where I met Professor Julie Cornfield and Dr. Suren Ponratnam. And the only reason I started working on this intraocular lens technology was because Dr. Julie and Dr. Ponratnam were such a fantastic people. I just trusted them that, okay, if these people are my co-founders, we can achieve anything. And I think part of it has come true. At least we have now reduced or removed the scientific risk and going into human clinical studies. So my motivation is not as exciting as Parul or Sachin's, but it was simply going with the flow and doing really what I wanted to do. So it could have resulted in a regret. Many startups fail. But fortunately, it hasn't till now. So,
thank you, Tanuj. Over to you, Jayant. So, Jayant actually was in the pharma industry, and really, I think, enjoys being an academic, right? So, his way of thinking is very different. So, let's hear from him what is the motivation behind the startup. How do you compare, the worlds of academic and the world of entrepreneurship, and how are you marrying the two?
I don't take anything serious, I think. Neither the academics, neither the company, neither the technology. That's my key strength because I know what not to do. I think I was looking for a day when this BIRAC grant came in, and I was actually unemployed because I fired myself from Piramal 2 times. So not a pharma, but I think a drug discovery, what Parul was saying about. And realized when I had no job that could I create my own job, because there could be many jobs, there could be so many opportunities after Humboldt and other fellowships. Dr. Badigar sir is nodding there. And having seen actually how the failures translate in spite of great ideas, in spite of very capable scientists, in spite of problems that what you see in a society. But I take my own problems. Can I create my own job? Can I create other scientists, a space for them to be engaged into a science world? That's how I look at it. And therefore, I looked after all, material science, polymer chemistry, clinical science, all postdocs. One fine day, I realized what is the most important challenge that material science can actually evolve into the, say, medical science or clinical science, especially in the diagnostics, especially in, finally impacting cancer patients' life. Because last 55 years, 60 years, we have spent trillions of dollars, but metastatic cancer patients' overall survival has been enhanced only by few months. And the problem is actually early detections so that. So I realized if I would design materials which would be the most impeccable in picking the rarest of an opportunity, and that was the point. So I look for a challenge, scientific problem of a translation which will take 10 years least. It should be first time, at least in this country. It should probably bring cost to 10 times, but should have a sensitivity double than what actually big companies has, and probably a success of only, say, below 1%. So that's how I take life of a science.
The scientific challenge being important, and your team members help you keep you focused on the business. Probably.
They say that you say it typically, I'll refer you, that I'm bad businessman, but today I'm meeting somebody who has, people have actually been very kind to spend money, time, give job. I think all high peers, Dr. Vadgaonkar sir here. I think I do not go behind the fund. I don't raise the funds, but fortunately last 2 years I can share here that about $4.5 million has come to me without actually writing a proposal or defending a grant. And that's the key, because I would like to say, I think all, Tanuj or Anuya, is everything is reviewed by the fund, those who give you funds, either the family or friend. They see your seriousness. How do you take actually, and the risk is actually if you are, not serious about your own, success. So there are two sides. Sure. I am probably bilateral here, but that's the key.
So Anuya, over to you. We're going to run out of time. So if anybody has any questions, please put your hand out so that I notice. Anuya, I think you had the unique opportunity in this at least panel of, spinning out a company. And you are aware deeply of both the academic world and the entrepreneurial world. What are the lessons learned? And are problems looked at differently? Problem definitions done differently? Is the approach to solutions done differently? Are the priorities different in these two worlds?
There's definitely a difference between how the problems are looked at in the startup industry versus how you look at it in academia. I think in academia, when you're looking at problems, the interaction with the customer is fairly limited. So understanding how your technology is going to be used by the person, or what are his key concerns, what are the pain points, are you solving all of them, probably does not reflect as much in the work that you do. But when you develop a product, I think there are different angles to the way it is looked at. For example, if you're developing for a medical professional, you need to look at it from the orthopedic surgeon point of view or the wound dressing point of view, that how is he going to do it? Are you making it easy for him? Are you improving the quality of life for the patient? From an investor's perspective, he doesn't care about what technology you're using. All that is looked at is whether it's going to perform better than what are the existing alternatives and can it make money. So there are various different ways at which you would, so the way you choose a problem in an industry versus way you choose in academia, I think is fairly different. Yeah.
Okay. I think since time is short, there's a question over there. Can I request, if you don't mind, can I just... Okay, good,
Hello.
good, thank you.
Well, each one of you had dream and which made it true, and you have ignited the minds of young and old, everybody. Now, India cannot become a developed country without women's participation, so Parul and Noya really shown the way. What is your message to the audience, particularly male fraternity, that how girl child should be developed, their mind should be developed? We have to produce entrepreneurs.
By the way, the question was to address, give your points for the male fraternity, okay? So over to you, Parul. But you can, of course, take a wider swipe at it, yeah.
I think I absolutely agree with you. And for me, equality and diversity come together. So if I have to give some advice to the male fraternity, it's just that just don't, when it comes to science, when it comes to brain, when it comes to entrepreneurship, there's no gender role. You just, the same advice that you would give to your boy child, you should give to your daughter. And you should just encourage them to do what they like, pursue their passion. Just give them this advice, everybody is going to do well. Everybody does well when they work on something which they absolutely love, irrespective of gender. That's the only advice you need to give.
Anuya?
Yeah, I absolutely agree with what Parul said. I think I come from a family where I had never faced any kind of a differentiation between how my brother was brought up or what I was brought up. And I think if that kind of this is given on, same goes with my husband. I mean, he's or your partner who has to be extremely supportive of what you do. So I think if that kind of an environment is given, then the sky is the limit. It's up to you to decide what you want to achieve. So I think the opportunity to choose and define what you want to do should be given to all equally.
There is one other question there, but can I just add one more point, since we have a large audience out here. One of the biggest challenges for women to succeed is breaking into networks, okay? And there is all said and done an old boys club, right? So it's important that seniors, people who are in the field should take extra efforts to open doors for women entrepreneurs. And that's something which is very, very important as well. So I'll leave you with that out. Your question, please. Yeah.
Okay. Normally, whenever startup approached, so
Can you
he
keep
or
it
she
close?
needs seed fund. And getting seed fund, there are multiple entries like grant, equity, then debit, and debit, convertible debit.
Yeah, what's your question?
Yeah, so my question is in perspective of incubator as well as startup. So other than the grant, because mostly startup require the grants, they are happy to have that. Other than grant, which is the best way to get seed fund?
Sure. Maybe Tanuj, you can take it on first, and then Sachin. Yeah.
So grants, of course, as you said, is one of the avenues of raising early risk money. Other than that, raising seed money is through, there's a lot of angel investments who would take early risks, okay? So you can't get all the money you need for completing your product development at one go. So you have to define the goals which you want to accomplish using the money which you are asking from an investor. So this can be either angel monies, individuals, your own personal investments. So when we started, we put some of our money plus some money from venture center in the form of equity. And I think that was a small amount of money, and I think till now we have raised both in forms of equity and grant more than $4 million. So it has to be built gradually. And if you look, I think Venture Center's website has a comprehensive database on where you can raise seed money or pre-seed money, Series A, pre-Series A, Series B. So that's probably you could have a look at that.
Yeah. Over to you, Sachin. So if you're interested, you can look up funding.venturecenter.co.in. There's a database of all funding pockets. The earliest part of highest risk is usually grant funded, and then it gradually increases to moves to little. But I think Sachin can talk about it.
Yeah, I think I very much agree with what Tanuj has said. So these are the major avenues. But for our case, I mean, I would advocate that if you have the potential to, fund your company in the initial years through grant money, nothing like that. Like, for example, we've raised around 4 million, out of which only 50 lakhs is seed investment. So I take a different opinion that until you have scope to raise money through these agencies, which of course come at an early risk. Of course, if you raise a private investment at a very early stage, you would lose a lot of your company. So you have to take a very sharp call onto if you have other ways. But yes, what Tanuj pointed out, like angel investors, your own money is something which, other than the incubator seed funding schemes, is always available.
Yeah, last question, if you're there, I think over there, please.
Yeah. So while all of you have done most of your work in association with NCL and Venture Center, and when we want many of such things outside, and when we are inside, we don't see what is that makes this happen. So, like, what are those unique things that you observed in this ecosystem that that are not there? So we are not having so many venture centers like systems elsewhere. So those unique points, I think, many people are from outside other institutions. I think we need to tell them what is this system that creates such an environment for you to kind of grow or take these risks.
Yeah, maybe I'll ask Jayant to address some of that.
I think when you are most desperate for funds, nobody gives you funds. And I think that desperation is probably a minus point of most of the early startup companies. I think the beauty of this place is that there is enough science, right? Great science, right? Dr. Lele, yourself, you have been actually our high PS. So this is the best environment probably for a startups company. But how does investors look when you are not associated with this kind of an institutes is actually they feel the risk is lesser because you're not entitled for a lot of administration work, a lot of scientific responsibilities, PhD students. So I think it's entirely, I look at it, if you are an independent technologist, it is better to have minimal associations, and that's what is, I think, a de-risking to the inventor. So any researcher has to actually look this kind of an, you cannot actually ride multiple, chairs, which I do. Of course, I have part of several, associations. But I think this is what is the strength of this institute, which can nurture innovations greatly.
Do you want to add anything, Sachin? Yeah, and we'll close after that. Yeah.
So, Iman, when I was a student here, the thing that I liked here was the crosstalk between different departments. If I come from a background in biology, I had Dr. Prasad, who was a good scientist in nanomaterials. So he also works as an advisor for us. So we are very grateful for his inputs. But in general, I think the crosstalk was very important for us to get started to a point where the problem was, of course, mass problem, but only with one set of skills, it was never possible. One thing which also I feel is that maybe in technical institutes, the knowledge of business is really important. In the journey of module innovations for like 7-8 years, what we realized little late, more deep into science, that ultimately what matters is how you translate that into business. And as Dr. Anuya pointed out, that investors want how fast you can multiply. So that is something which should be a part, or if that is included. Of course, we have Venture Center here, but for other institutes where this is not happening, crosstalk is not happening, that's going to be a key.
Okay, so we are ready to close now. Please, before we thank, okay, so Ashish has a question there.
No,
I can't say no to Ashish, so.
I don't want to ask a question. I just want to add on to what people have said in response to Prasad's question. I think what is most important and not said anywhere is that you can have brick and mortar buildings, but that is not enough to create the ecosystem that we have at NCL. You need visionary people like Dr. Shivaram and Dr. Premnath. Please join me in it is their vision and their leadership and their very systematic approach that has really built the foundations of this ecosystem. And as entrepreneurs, you need that support. You need to be able to have people around you who can help you both in terms of systems and in terms of that positive attitude, which I think is a very, very critical part of the ecosystem at NCL.
Yeah.
Without that, as I said, you can just have bricks and mortar building, but not that kind of support. So I think that is not said anywhere. It's very important that institutions are built by people.
So, Ashish, I second you on that. Actually, you stole a little bit of what I was going to do. So, by the way, champions are extremely important, okay? You cannot do any of this without champions because you're looking ahead in the future. There's a lot of uncertainty. You don't know whether things will work. And we are very fortunate that we have had champions in this community. Of course, Dr. Sivaram, I think we should give a round of applause, but also people like Dr. Mishra here, right, has also done wonders in other institutions. So I think the polymer science community in India is blessed. We should make the most of it. It's time to strengthen it and not necessarily, let it become just one other community out there. So let's hope we can do that. Please thank the entrepreneurs here for their journey. And, let's wish them all the best in their journeys. Yeah, thank you. Please.
Thank you, Premnath, for moderating this wonderful session, and thank you, panelists, for letting us know your journey and whatever the way you came to this and being an entrepreneur. As a token of appreciation, we would like to give you memento from the organizing committee.
Yeah, I think Mike you might need. That's a book from Mr. Satyen Daga out there. Yeah. Also, besides, of course,
You have to come that close.
yeah,
You
come.
can see two women, one giving the award to the like gift to the other. So we have more.
Can you come here? Yeah. Please, next. Thank you. In the bag, maybe. Thank you.
Yes.
Thank you very much.
And we missed giving the memento to Guru. Where is?
Guru is there on the left.
Yeah, Guru, please. Keeping time, we love.
I think you can do it in front, huh? There, because it comes well with the picture.
So that brings us to the end of the first session. There is tea outside, there's tea break. But we would like to take a group picture. So before the tea, if all of us can go to the front of the main building, it would be nice because Dr. Shivram is there, Dr. Premnath is there.
Hello. We welcome all of you for the second session in the morning. So the chair for this session is Dr. B.L.V. Prasad. Dr. B.L.V. Prasad is the director of the Center for Nano and Soft Matter Sciences, CENS, Bengaluru. So he'll preside over this session and he'll do the deliberations.
Okay, good afternoon. We are running little behind schedule, so let us quickly start. I request all of you to please settle down. And also, whoever is going out of this door, at least close the curtain in order to stop distracting the speaker. We have two speakers in this session, session 18 of this SPSI macro. They are Dr. Ajay Ghosh and then Professor Jay Kannan. The first speaker is Dr. Ajay Ghosh, but before I start, let me also offer my respects. Dr. Shivaram is not here, and best wishes to Dr. Shivaram on his 75th birthday. Dr. Ajay Ghosh is the first speaker. I don't think he needs any introduction, and then because we are running short of time, let us not go through any introductions here. And he is going to talk to us on two-dimensional polymers of ionic covalent organic nanosheets. Dr. Ajay Ghosh, please.
Thank you, Prasad. Good morning, everyone. It is always a pleasure back to NCL, especially to this auditorium here. I have given several lectures. I'm sorry that I missed the last 2 days lectures and deliberations due to some other assignments. But I'm sure that a lot of things must have been said about the polymers and polymer-related chemistry, physics, and engineering. I'm also very happy because this conference is also dedicated to Dr. Shivaram. We were celebrating his 75th birthday and birth anniversary. And in fact, I was fortunate to give a lecture before, sometimes back in Bombay where we were celebrating his 75th birthday. In fact, he is one of the motivating figure for many of us to pursue science in general and polymer chemistry in particular. So, congratulations and best wishes to Dr. Shivaram. So I come from the down south of India, the state of Kerala, where we have a CSIR laboratory, CSIR National Institute for Interdisciplinary Science and Technology. And this is the main building of the institute. Some of you may be aware of this institute. Those who are not seen the institute, if you happen to be in Kerala, please do visit us. Of course, just recently I superannuated from the institute, but still associated, maintaining the laboratory and my activities. The main research interest of my research group at this institute is on soft material research. Of course, I started with a polymer chemist working in photopolymerization, photodegradation of polymers and many other kinds of polymers. Later, moved on to supramolecular chemistry and working on supramolecular polymers, molecular assemblies, architectures, so on and so forth, making use of many different kinds of pi systems. I'm sure I need not give you an introduction about the pi systems and its relevance in the new generation technologies. By virtue of those interesting electronic and optical properties of pi systems, they are active component in many kind of devices. But we make use of small pi systems And we explore the possibilities of non-covalent chemistry to create different kinds of soft materials like organogels, many different kinds of supramolecular architectures, and many of them are stimuli responsive materials which respond to various kinds of stimuli. We also make use of many of these fluorescent pi systems as molecular probes and sensors for diagnostics applications. And lately, we also entered into coordination polymers and covalent organic frameworks, of course, bringing in some components of supramolecular chemistry into it. As I mentioned, to this audience, this is a chemistry audience, so you are all aware of the contributions of chemistry for more than 100 years to the society and what polymer chemistry has done, and what are the other associated consequences. So scientists as a whole trying to further address those issues. So many of those polymers which we are talking are conventional polymers, but at the same time, polymer chemistry over the years was, continuously undergoing changes and evolution, and many new generations materials have come up in many different ways. So we can generalize all those polymers to distinguish from this class as non-conventional polymers. And when I say non-conventional polymers, they are of many different types. I am listing 2 or 3 of them, but you can add several more into this, which may be in many ways different from these class of polymers that we usually address. So one of them are supramolecular polymers. I have noted that there was a lecture the first day or the second day on supramolecular polymers. I also used to talk about, because this is one of my favorite topics in previous macro conferences, I address many of these. But at the same time, there are other kind of polymers, for example, metal organic polymers, metal organic supramolecular systems, and now recently covalent organic frameworks, the so-called COF. And in this point, you may remember that there are many other similar class of materials started with MOF, metal organic frameworks, and then now covalent organic frameworks, and then covalent organic nanosheets. So I will briefly take you to some of these as an introduction, and then I will move on to the topic which I want to address today to all of you. So supramolecular polymers, this is one of the latest development in polymer chemistry. The advantage of supramolecular polymers is that the building blocks can interact to form polymer chains using weak non-covalent interactions, mainly hydrogen bonding and other secondary interactions. But to use such polymers, you have to make them relatively stronger under different processing conditions, which is the greatest challenge that scientists are facing. And they started with, the famous Jean-Marie Lehn, and he introduced these kind of systems. He has shown some beautiful structures on, different types of supramolecular polymers, and later, Mayer and Doven has demonstrated a beautiful example of supramolecular polymers by invoking the power of multiple hydrogen bonds, stable multiple hydrogen bonds with a very strong association constant. So when you have prepolymers which can enter into these kind of interactions, which can lead to kind of one-dimensional polymeric chains, having properties which are similar to many other kinds of conventional polymers that we address for many different kinds of applications. So you can see from this picture, it shows like a viscous liquid, which is the prepolymer, but on further polymerization, you can make it like an elastomer, an elastic solid. And the advantage here is that if you want to reprocess this or remold it, you can always heat it without decomposing the monomeric unit. So you can get back the monomeric unit and then you can remold it. And this is very, very relevant when we talk about reprocessing and recycling of polymeric materials. But at the same time, there are many inherent limitations for being a viable material for many practical applications. A lot of things are going on, and but there are certain niche areas wherein such kind of polymers can come in and play an important role in terms of technological developments. The other class of polymers are metallo-supramolecular polymers. This is a report way back from our research group in collaboration with a group of people from Japan. Actually, this originated because, at that point of time, we had many interest in highly colored materials like organic functional dyes. And many of those functional dyes can be further modified structurally and can be used as active building blocks to create various kinds of supramolecular architectures. So this is one of the earliest. It came in 2007, and we made a metallo-supramolecular polymers which respond to metal ions, and it changes their optical properties. It just happened like that, and then at that time there were not many reports in the literature on different kinds of metallo-supramolecular systems. So we just left it, but later we found that a lot of interest are being generated in this. So we started reopening these kind of problems, and we published a few papers on metallo-supramolecular systems which can form soft materials like gels, and also which respond to various kinds of external stimulus, like, say, for example, light or mechanical pressure, etc. And interestingly, many of them, when you do certain kind of stimuli responsive changes, it can also influence upon the overall hierarchical structures, and then you can have certain kind of control on the eventual structure that evolved from these kind of molecular assemblies. So here is an example where you can create two-dimensional sheets which undergo further assembly to form certain unusual structures, like, for example, cabbage, flower-like structures, and other star-like structures, where we don't have much control on this in terms of their size and shape, but you can bring in some kind of control to some stage. And then we were continuing this on this particular topic, and this was a paper which was published sometimes back in Nature Communication. This is Rahul, who was my PhD student, and he is is one of the brilliant students that I ever had in my group. He has made some interesting supramolecular systems having pendant photoresponsive system. Here it is isobenzene pendant, and he has shown that you can create one-dimensional metallo-supramolecular system with hanging photoactive pendants, wherein you can use host gas chemistry. So he can complex with cyclodextrins, and then he can release cyclodextrin against, in response to temperature in a stepwise manner with a lot of control. And so he has shown that, he can stepwise release one at a time these cyclodextrins, and he can also, complex it back one at a time with a lot of precision using temperature as the external stimulus. And this is one of our recent report, again in metallo-supramolecular polymeric systems, which shows a sort of reversible thermochromic color change, which can be used as a logic operator. So there are small molecular systems which are being used in logic operations, but large molecular system, macromolecular systems, especially these kind of metallo-supramolecular systems, are not being used. And they, at different conditions, especially with temperature, they respond. And here is a composite gel which is prepared from these kind of chromophoric systems with other polymeric gels. And then you can cast it on different surfaces which then change temperature, color with temperature on a reversible fashion. Now with this kind of introduction, I will now invite your attention to the topic that I would like to focus on the next maybe 15 minutes that I have. So that is on covalent organic frameworks, which is a very, very active area here. And earlier here in NCL, I think Rahul Banerjee was working in, he's one of the most active figures in this area from India. And so sometimes back we got interested and we also collaborated with Rahul on this. So covalent organic frameworks can be considered as a kind of polymeric system. And if you look at the Wikipedia, you have the simplest definition of covalent organic frameworks as a class of materials that form 2- or 3-dimensional structures through covalent reactions between organic precursors to form a kind of a network. Or, this can be 2-dimensional or 3-dimensional. So for example, you can bring in the so-called reticular chemistry with a suitable component like this. But you can see, depending upon the topography of the building blocks or the monomeric unit that you use, you can articulate many different kinds of structures, which is called the reticular chemistry. And if you can bring in a little bit of topological control on reticular systems, you can create a kind of more crystalline structures. They are called, covalent organic frameworks. But as it is mentioned here, they may not be always crystalline materials unless you have some kind of a topochemical control. In many cases, they are, amorphous kind of 2-dimensional sheets, which can be considered as supramolecular or, 2-dimensional polymeric sheets. Of course, these kind of polymers, they lack many properties of conventional polymers, like, for example, their solubility in different variety of solvents, and the signature of many of the polymers, that is the viscosity, and many different kinds of properties are missing. But in many other ways, they behave like polymeric materials. So here is one example, one of the very best example from Omar Yagi, who is very famous, known for metal organic frameworks. So this is phenyl diboronic acid reacting to form a kind of a hexagonal network. This is one unit here which extends from all directions to form a sort of a 2-dimensional sheet-like structures. And when they form, they further, self-organize these sheets to form a kind of a 3-dimensional materials. And then you can exfoliate them under different conditions. And now from COF, you can have subdomains of different types of materials. One among them is the ionic covalent organic frameworks, that is ICOF. So the importance here is the material is ionic. So, and then the COF may be bulk ionic materials, and when they exfoliate, you get ionic covalent organic thin nanosheets. It can be a single layer or bilayer or multiple layer nanosheets. And the role of the ionic group is that, when you put it, there will be a lot of, repulsion between the ionic groups, which will help them to do a sort of, self-exfoliation. So you always have the advantage of getting exfoliated single sheets or double layer sheets in solutions. And they are more compatible with polar solvents because they are ionic. So there are many examples in the literature on various kinds of studies are reported in the literature. And then we thought of making some kind of icons, that is ionic covalent organic materials and ionic covalent organic nanosheets. And so we were looking for the suitable building blocks, and at that point of time we found that this has a lot of biological significance and a lot of other significance from the materials viewpoint. This is familiar to many of you, ethidium bromide, which is a well-known DNA intercalator. So when you deal with, you have to be extremely careful. It is a highly toxic material. Don't allow to come in contact with your skin or in any other ways. So they can go and bind in the DNA grooves, and then you get intercalated DNA. But if you look at, there are two amino group over there which you can subsequently use for doing further chemical transformations. And then we have taken this as our building block, and then we have used many different kind of chemical entities to make different kinds of covalent organic frameworks. So one of the example is this triformyl fluoroglucinol, which is known to efficiently react with this amino group to form these kind of hexagonal kind of cyclic macromolecular systems when you do a solvothermal synthesis. So based on this, sometimes back we published 2 papers, and the work is done by Dr. Arindra Mal, who completed PhD sometimes back from my group. I'll be discussing these 2 papers a little bit more in the next maybe 10 minutes. So this is an example for an ethidium bromide based ionic covalent organic nanosheets. And so this is the structure of those. I think something is wrong with the slide here, but I will, I think one frame is missing somewhere in this one. So doesn't matter because the AFM image is not here. And we have a clear AFM image on and TM image on the structure of these ionic covalent organic nanosheets, and they shows their characteristic structural features. And then what we have done is because it has this ethidium bromide moiety in this, and what we anticipated is that when you take DNA, different types of DNA, and try to react with this, probably this ethidium bromide core can interact to DNA to form many different types of hybrid materials, and we were curious to know what kind of properties that you can derive from such hybrid materials. So here is the interaction of DNA with such kind of ethidium bromide based icons. And so you can see here, this is the AFM image of the hybrid material. And you can see the height image here, it is something like 30 to 40 nanometer. That is what on this marked area, this is the sheets, hybrid sheets that you have. But since the previous image on the virgin material was missing, I would like to tell you that the height of that covalent nanosheets were something like 1 to 2 nanometer. That was the height of this. And from that 1 to 2 nanometer, when you interact it with DNA, you get this kind of from 2-dimensional to 3-dimensional structures with a fairly high height of 30 to 40 nanometer, which means that those single sheets or double sheets were interacted with the DNA, and the DNA facilitated further stacking of this, and you are getting stacked, restacked, or reassembled nanosheets. And here is the scanning electron microscopic picture, which shows a sort of an unusual layer type assembly, indicating that this insert shows the side view of those organized assemblies of these nanosheets and DNA. You can see layer by layer organization of this, which justifies this height image of these materials. And to have more details on the nanosheets, you can go for the detailed TM analysis. And you can see here that this is the TM image, and here is the the SAED pattern, which shows some sort of crystallinity, which indicates that the reassembly with DNA is more organized and it gives sort of a crystalline order to this hybrid material. We do a lot of studies, particularly we investigate the optical properties, and we are particularly interested in the fluorescence behavior of many of those materials we synthesize in our laboratory. So we immediately look for what happens to the fluorescence of these materials. So what you see here is this, this fluorescence is the fluorescence of this material, single layer or bilayer icons. But the moment you interact with a Kalf-Thymus DNA, which is a double-stranded DNA, you can see that the fluorescence is significantly quenched. That is because of the packing of these various layers into a three-dimensional structure. There is a sort of called a self-quenching, or due to other physical phenomenon, the fluorescence is significantly quenched in the composite material. And then this one is the, this one you see here is the fluorescence of this material in the solid state. This is in solution, what you see here. So this is in solid state. In solid state is completely quenched. See, most of these molecules in the solid state have highly quenched emission. But the moment you add DNA into this and make a composite, you can see that at least 2 to 3 times enhancement in the...
Dr. Ajay Ghosh, I think 3 minutes, can you close?
Oh, okay, fluorescence. So then we looked at the zeta potential, and then we did a lot of studies on these kind of composite materials, looking for what kind of applications. So this is the interaction of this ethidium bromide based icon with the DNA. As I mentioned, once you add DNA, you see a significant color change with as the concentration of the double-stranded DNA is increased. And you can see that it is very, very selective. These are some of the other compounds having some sort of phosphate group or ionic groups which are non-sensitive to these composites, these icons. But when you have DNA, especially single-stranded and double-stranded DNA, you see some response. But the response is much, much prominent in case of double-stranded DNA. So then we thought that this can be used as a sensor platform to distinguish between single-stranded and double-stranded DNA. So this is the response, the ratio of the fluorescence response against various kinds of DNA of various chain length. So what you see on the top of this figure here is these are the fluorescence response of these icons against a double-stranded DNA of different chain length. Whereas the corresponding single-stranded DNA shows the least response. That means the single-stranded DNA is not interacting strongly with this kind of icons, but the double-stranded DNA interact very strongly, and they induce a sort of stacking of all these layers in a crystalline fashion, and that enhances the fluorescence of this, which can be used for the detection of double-stranded DNA from a given solution. Then subsequently we expanded this, and since a short of time I am not going to discuss these things in details, but what I am showing is that we can make different type of such ionic covalent organic nanosheets which undergo self-exfoliation in this and can be used for various kinds of applications. You can do a lot of chemistry with this, for example, whether it is supramolecular chemistry or non-covalent interactions. You can use those kind of interactions to create different kinds of hybrid materials having intriguing properties. So with this, I don't have time, so I will close it down by saying that you can design different types of materials which can respond to various kinds of external kind of analytes. Or here, for example, this is a system, supramolecular system, which can respond to different colonies of bacteria, different types of bacteria, gram-positive or gram-negative, because these kind of materials have a lot of ionic groups on that, and the ionic groups are known to interact with the bacterial cell walls, and it can kill them. So there are a lot of scope on these kind of materials which are antibacterial and which can respond to such kind of microorganisms at different conditions. So we have shown that they are highly sensitive to bacteria, and we are now looking for more efficient systems which can be used as antibacterial materials as a part of a course materials. So this is the overall process happening. When you have the icons, they self-exfoliate in water or other solvents, giving you a large number of single layer icons, which can then interact with bacteria, break the cell walls of bacteria and kill them. And you can do it reversibly, invoking many different kinds of supramolecular chemistry into that. With this, I would like to close the talk by thanking all these people, particularly this gentleman, Arindam Mal, who did all this work, and also all the funding agencies, and thank you for your kind attention.
Sorry, we had to rush you a little bit.
I know that we started late. And
Okay, so I think again, since we are little running behind the schedule, one or two quick questions. Yeah, there is one question there. Can somebody pass on the mic? That corner. Dr. Nityanandan, can you guide the volunteers? You raise a hand, right? Okay. I saw you raising a hand. Yeah, there is one question. Dr. Shahjahan.
Are these material porous in nature?
They are porous, yes. Because they are porous materials.
Does this porosity decreases compared to normal COP materials?
No, that depends upon what is the kind of cyclic ring that you are creating in this. So they are you look at the structure, that they are large.
For example, if I have a similar structural entity except the ionic part, you think the porosity would decrease? Because the one of the application of COPs are on gas absorption and many
gas
other
absorption. So these are, of course, porous materials, but the porosity is in the nanoscopic porosity. And in the bulk material, probably the porosity is you cannot control those porosity when you make these stacks, because the stacks have to be highly crystalline to have this kind of porosity uniform. But what you can do is you can control the porosity by choosing the appropriate building block. For example, if you keep on increasing the length of the building block, you can design the kind of porosity that you required in a molecular level. So,
okay, I think we will close now because we are running behind schedule. It gives me a great pleasure in giving this memento to
Thank you. Thank you very much.
Sorry.
Okay, yeah. We move to the next talk, which is to be given by Professor Jayakannan. He's from ISER Pune. He also spent some time earlier at NIST Trivandrum before moving to ISER Pune. Professor Jayakannan. He's going to talk us on tweaking polymer topology and breaching bacterial cell membranes. 22 minutes, I will give you an indication to close in 3 minutes.
Thank you. Thank you, Prasad, my good friend, for the introduction. So first I'd like to thank the organizer, Dr. Asha and Dr. Sami Chikadi, for the invitation. I think this is the last technical talk on polymer science. I think till the next macro we all meet again. So welcome to the last meeting. And first I'd like to thank Professor Sivaram. He has become 75 years young again and going to be 76 years today. And from my institute, from Indian Staff of Science Education Research, and Professor Shivaram contribution is very immense from starting the institute from the scratch where we are right now we are here. And on behalf of all my colleagues at the institute and my department of chemistry, so sincere thanks to you for your all contribution to build ISR, what we are ISR today. Thank you very much. So with that, I would like to go to the technical part of this today's presentation. So this particular topic, how we can use polymer topology, basically you can make a very large star-shaped polymers, and how we can use for breaching the bacterial membrane, and where we can also look into the antimicrobial properties. This particular topic we have been learning for last 5 years, and this is the first time I is giving on this topic to the polymer community. Okay, so why do you want to work on these polymers? What is the importance of antimicrobial research? There are two worlds in this earth try to control our life. One is the thing, if you look at the size of, something wrong, okay. If you look at the size of some of the microorganisms, if you strictly put a scale here, that this micro world actually try to control this macro world where we are in the meter range. And this micro world, it can control such an extent that it can keep us locked inside a house for suppose 2 months during a pandemic, viruses, bacteria, anything. So basically the macro world takes 2 years to understand the system, try to make the vaccine, try to control the micro world. So the micro worlds are very powerful. And this micro world, how it is going to influence on your healthcare of the human population. If you look at that, this is the prediction from the Wellcome Trust, very well-known funding sources from UK. This is a 2014 data, but it's more or less the scenario is same. This is up to the 2050 prediction. Okay, if you look at that, among all the disease which is going to influence on the human health care, I think antimicrobial resistance is the top. So enough people die in a road accident, that's a different scenario. But antimicrobial resistance is going to be the dominating factors which is going to influence on the human health care. The second disease which is also equally going to influence is cancer. That is the reason many people in biomaterial science, either they work on antimicrobial resistance or cancer related problem, because this is very important factor. And then if you look at that, how this disease is going to influence on the global GDP. So this is the average expected global GDP, about $100 trillion in 2050. So definitely the way at which we are growing, in 2050, India must be at least $10 trillion economy. A $10 trillion economy, this antimicrobial research alone can bring your GDP by 10%, which means the $10 trillion economy in India, if you grow the same rate, our GDP can be brought down by 20% exclusively by antimicrobial resistance as well as the cancer disease. So that kind of, for our population of 1.4 billion population, we cannot afford to lose the 20% of the GDP, okay. So basically, many research activities globally, not only in India, people are focusing on how you can keep your population so healthy so that you actually generate enough wealth, okay. So this is the scenario where people are looking at the biomaterial science, okay. So keeping this focus, and in my research group, last 12 to 14 years in ISR Pune, we actually work on amino acid as a raw material source. We have a chemistry, it's a solvent-free melt polycondensate chemistry. We can apply that to amino acids, we convert them into a suitable monomers, and we can make a polyesters or polyurethanes. This I call non-peptide analog of amino acid starting material, okay. So this is something which we do, and we also use these polymers, use the nano dimension, so we can actually use for a drug delivery, and this kind of application we are exploring in the group. Recently, last 2 to 3 years, we have been working on how we can use this amino acid as a source. We also come up with a steric, using a steric bulky appropriate group, where we can actually use this chemistry for variety of inaccessible polypeptides, which is not typically accessible based on the serine and cysteine kind of systems. And these polypeptides, we can also use it for a biomedical application in future perspective. This is another synthetic approach which we are working on. But today I am going to talk to you on how we use a polycaprolactam platform. There is another biodegradable polymer, how we can control the topology of the polymer, whether you can use either linear or a star-shaped analog. If you look at these two structures, the number of units which essentially have exactly same, but the topology is completely different. How they could actually play a very significant role on the antimicrobial research, which I am going to talk about today. And these similar kind of structures, we also indeed explore for how you can selectively target for delivering certain cargoes to the brain model also, so which are working on, but I will just today summarize our contribution on this particular area. So if you look at this data, this just came in 2022 in January, And this is the kind of bacterium which can actually influence on the human healthcare. So we think that E. coli is a very healthy bacteria, but once they get mutated, they are the most affecting bacteriums in actually in our healthcare industry. The next one is SREs, and these two are the two dirty bacteria which can actually bring down your healthcare much more drastically. And this paper appeared in Lancet in 2022 this year. You can look at that how a gram-negative bacteria like E. coli, when suppose they undergo a kind of a resistance to the kind of a treatment which patients take, 70 to 80% of the patients actually develop high resistance towards this kind of the bacteria. So that your drug become ineffective, and that severely influence a country like India, okay, because we have a lot of tropical kind of weathers, and similar to that of the SRAs also. So basically these two bacterium heavily influence on our country, okay. So another dimension to the reason for why do you want to do that. Okay. So what are available for us to treat any infected disease? So there are a lot of clinical antibiotics which are developed in the market, and we are using like penicillin to many antibiotics, and they have a very clear, well-defined pathways in which they actually affect in which way you can actually cure your infections. And now these kind of drugs, the clinical antibiotics, they outdated very fast in your by microorganism, because they are microorganisms very smart enough to develop a perfect. They undergo mutations, they can actually develop a resistance to these most of the drug molecules. So within 10 years, most of these clinical antibiotics are outdated. Okay? So that's why if you go to the doctor, he will try with one antibiotics, then he'll try with the second, third, it keep on goes through that, because they are very smart. So these are some of the number of, suppose within a day, for example, a E. coli like a bacterium, if there is, when they split in 20 minutes, this is a 20 minutes a cycle for each bacterium to split. Suppose if there is a one bacterium undergo a mutation, and this bacterium within a day it can actually split into 10 to 20 power, 10 to 23, power of 23 cells, which is almost similar to Avogadro number of molecules which you are talking about. So many infected bacteria it can produce. Then you can imagine the resistance which they develop, which most of the time is antibiotics fail. Now the second generation antibacterial reagents are basically, they are like a large peptide analogus, which my colleague Professor Gopi also works in similar kind of area in ISR Pune, and these kind of a large amount of the oligopeptide are cyclic or linear peptides, and these kind of systems can adopt any of these kind of the self-assembled structures. All these structures are tried, everything works depending upon the need. And another new generation of antibiotics which are emerging, the third generation antibiotics, they probably based on a non-peptide beta based analogus. They could be a cationic surfactants like these, or it could be a small molecular amphiphilic peptides, like we have a few peptide unit which have put up a lot of amphiphilic units, or it can be completely a macromolecular system, a polymeric based systems, where you can have a strong acrylic backbone, where you have appropriate pendants are attached in order to have antimicrobial activity. So this third generation antibiotics provides very unique opportunity, because if you combine these particular systems along with the nanotechnology, where you can actually make variety of self-assembled structures like micelles and vesicles, and these structures in principle you can actually encapsulate this kind of the clinical antibiotics. So basically you can bring two different systems in a single combination, where you can have a maximum efficacy. So that is the advantage of one would using this kind of the surfactants or cationic systems or a polymer system, where you can actually employ the nanotechnology, which has some limitation in the case of the antimicrobial peptides. Okay. So now how these antimicrobial peptides or polymers, they basically work on the bacterium. So these are some of the mechanism which is very accepted in the case of the antimicrobial research. So they go through this, basically the antimicrobial peptides are polymers, they actually attack on any of these membranes or bacterial membranes, and they go certain process where the membrane is completely, the stability of membrane is compromised, and the bacterium undergoes actually death. Okay. So now especially in the case of the antimicrobial polymers, if you look at that, most of the polymers are based on the polypeptides. It could be lysine or histidine, or it could be arginine based polypeptides, or it could be polyacrylates, polycarbonates, even several area of conducting polymers are also known to do this kind of job. But if you look at that, most of the materials which are so far studied, nearly 90% of non-biodegradable in nature. Okay? So as a polymer community, one would actually seek that for a biomaterial application, if you do a biodegradable polymer, that will be the best way to move forward rather than actually getting stuck with the non-biodegradable systems. And the second thing, that suppose we are working on a biodegradable polymer, your research should not be like you are adding one more example in the literature. Okay? So we have to do something substantial more than that. So one of the limitation on these systems is that you do not know the mechanism at which how this bacterial cell membrane is influenced by many of these structures. So it's also equally important, one should also put enough effort to understand the mechanism as well. Okay? So these are some of the gray areas in this particular domain. Okay? So many people have a different strategy, but we have a little bit different strategy than others. So since we want to work on that, if you want to move to the ideal antimicrobial polymers, then what are the criteria the ideal antimicrobial polymers required to have that? So one of the important things that in the event you try to inhibit the bacterium growth, eventually you come in contact with this kind of polymers with any mammalians, including human or anything. So when you such a polymer which you are going to deal with that, in the event you come in contact with such a cationic, very strong charged polymer, that should not become toxic to your human. That is the first important thing. Second thing, that suppose in the event, not only the on periphery of this coming in contact, suppose you take it by go through your bloodstream, and that polymer should not be a non-hemolytic nature. There should not be hemolytic to the red blood cells within your bloodstream. That's the second criteria. And the third one is that in the event such a polymer nanoparticle, we are using it for inhibiting the membrane breakage of any bacterium, like they are puncturing the bacterium. For instance, the bacterium has a lot of enzymes which are stored in your cytosol. They will actually come out. Okay? Once they come out, for example, the enzyme which is coming out from bacterium, it can actually be a counter-emulative. It can actually degrade the polymer itself. Okay? So which means use a polymer in order to inhibit the bacterium whatever they excrete, that can actually use for degrading the polymer. So that kind of approach also one can do, but then you have to deal with the really enzyme degradable polymers which can be non-toxic to cells. It should be non-hemolytic. These are two important factors. And the third thing, it should be lysosomal enzyme responsible in the event it goes into the mammalian cells, or it should be antimicrobial for doing this action, and whatever the excrete come out from the bacterium, this polymer also should be bacterial enzyme responsive. So if you have these multi-component systems, one can actually eventually go through the ideal antimicrobial systems which can be solve the problem in long term. So this is the concept which we thought maybe it's worth to explore in the literature. And then what are your choices? What are your choices for dealing with a completely biodegradable polymer? You don't have much choice. You have only few choices. For example, you can deal with a caprolactam. Okay, caprolactam is a very good system because it forms aliphatic polyesters, or this is a polyhydroxybutyrate. This is also now coming in the literature. And we have some polysaccharide or polypeptides or polylactates. So if you look at the biodegradation aspects under the physiological conditions, your polypeptides are champions. They can completely degrade. On the other hand, if you look at the environmental stability, polycaprolactam is a champion. You can deal with them in the lab much more easy, but the degradation is compromised when you go to the caprolactam systems. But that only apply to the fully unsubstituted polycaprolactam. The moment you deal with the very derivatives of polycaprolactam, you can actually bring down the degradability even less than 18 hours. Okay? Because the enzyme can the enzyme can basically adhere on the polymer chains, it can actually degrade. So if you have appropriate chemistry to do that, polycaprolactam is excellent material to work with that. And this particular concept we have been exploring for the past several years, how we can use polycaprolactam based systems for drug delivery applications, and also certain cancer specific model which we are developing in the group. So based on this experience of our in the polycaprolactam systems, now we have actually designed a polymer. The polymer has a polycaprolactam backbone with appropriate cationic charges which you can put it on the periphery, and this can form a nice 5 nanometer at very tiny nanoparticles which can form in aqueous medium. And this one we actually use it for checking various antimicrobial properties. For example, here we use both the colony suspension method and growth method, two independent methods. The data, I am not going to the details, they clearly suggest that these polymers are indeed very effective in acting on the bacterium. And if you take this, the E. coli, basically the membranes are negatively charged since you are using a cationic polymer, there will be a charge neutralization happen because of anionic cationic interaction that actually makes your polymer to bind on the membrane of your bacterium. You can use standard zeta potential measurements to in order to validate, and also you can look at electron microscope image how this polymer actually breaks your bacterial membrane. These are some of the indirect way of you can say that is the polymer indeed acting on your E. coli. And we also looked at that what would be the chain length effect on the effect of the bacterial membrane, and we found that 10 units of this polymer chain is enough. You only need not to make very high molecular polymer. Suppose if you make 100 units very high molecular polymer or 10 units, the effect is basically same. So even oligomers of 10 to 15 units is sufficient enough for this particular purpose. Okay, that's what we shown. And then we also looked at that how these nanoparticle actually influence if in the event it goes to your endothelial cells, and we found that these polymers are basically they are non-toxic to your mammalian cell lines, and also in the event they actually interact with the RBC, also we looked at that the polymers are pretty stable to your RBC. So they are not hemolytic, non-hemolytic, they are very stable. So these polymers are very unique. Now when we looked at that, okay, now we know that okay this polymer is working all right, but how do you test that the polymer really undergoes enzymatic degradation, we can prove the biodegradability. That is the question. So for that we taken a very well-known dye, HPTS, we could encapsulate it in this nanoparticle, and the size is still retained 5 nanometer. And then we use in vitro analysis. Basically we use a esterase enzyme, which is lysosomal esterase enzyme, which is abundant in mammalian cell. So when we actually use release kinetics profiles with this nanoparticle, we can see that in the absence of the enzymes, the particles are pretty stable. On the event of the enzymes, you can see 100% they undergo degrade, release your molecule, which means basically the polymer can undergo lysosomal enzymatic degradation. Similarly, we taken a bacterial enzyme like lipase, which is overexpressed in the bacterium. You see a similar profile in the lipase as well. Basically, we prove that these polymers are biodegradable. So even if it come in contact with the mammalian cells or mammalian cells, they can be degraded. Okay, so it's not going to be toxic in nature. So once we established these procedures, and then okay, now we looked at that whether really how these polymers interact on the membrane of the E. coli. So if the polymer interact on the membrane, now it will form a pores basically. Once you get the pores, you can use a secondary marker like propidium iodide. Propidium iodide marker, it can easily penetrate from extracellular to the entire membrane, so that once it goes, it can bind to the DNA of your bacterium. So you can use it as a model, you can do that. So we use a confocal microscope image in order to quantify in the absence of the polymer treatment, the presence of the polymer treatment, and we could see that the polymer actually definitely inhibit on the membrane, and then you can actually estimate it using how much you are co-localizing with the propidium iodide. Now we take this polymer, the polymer which is a luminescent loaded with a HPTS dye, and since it is a HPTS dye, the HPTS is not a non-antimicrobial, it's a simple polymer. So when the polymer, suppose the polymer binding on the membrane is a two-step process, not a single-step process. First, because of the membrane is a negatively charged, the particle is a positively charged. First, it has to come and adhere on a membrane surface. And the second is that because of these interactions and other hydrophilic, hydrophobic interactions, they form the pore. So the moment it forms a pore, you can actually stain with the propidium iodide. But if there is no pore formation, you cannot stain with the propidium iodide. So it's a two-step process. So if you want to monitor this, you have to do a real-time analysis, and that's what we did. We actually looked at this E. coli as a model, how these polymer nanoparticle can undergo this mechanism, and here I shown a confocal images which are actually recorded at various time interval, and you can clearly see that the dye basically adhere on the polymer of the dye basically adhere on your polymer at various time point, but propidium staining is exclusively happen at the higher time points. Basically now we can estimate it how many hours actually one required in order to make this polymer to be effective on the bacterium, and that we can actually estimate it. And also when you co-localize these two different probes, you can also look at that. You can look at that how a dead bacteria will look like orange because of the combination of the green and red signal, or the one which is not influenced still look like a green signal. So you can actually use a visualization technique. One can actually estimate it, actually quantify it, how much bacterium is alive, how much bacterium is dead. Okay, so this is the concept which you developed in the lab. Now, by doing this, what you do that basically indirectly you gain information that okay, my polymer is actually binding on the membrane. You cannot directly visualize the polymer by this process. So for that you need a higher level understanding required. So we adopted the concept of aggregation induced emission. Now actually we're making this polymer which are actually capped or are tagged with this kind of chromophores. When I have a tagged chromophores, now I can directly visualize my polymers adhere on the membrane. Now once the polymers adhere on the membrane, because of its antimicrobial act, it's like a gate. The gate is closed of the membrane gate. Once the moment your antimicrobial polymer act on the membrane, the gate will open. Once the gate is open, you have a pore. Once you get the pore, you can also use a secondary marker. So here propionamide is not useful. We use a secondary marker, basically crystal violet as a staining agent. The crystal violet exclusively it stain on this middle layer, okay, lipophilic layer. It will not bind on the side membrane. So now we have both a polymer as well as a secondary marker in the membrane. So you can use a FRET as a probe. You can actually visualize and quantify it, whether you can have a higher level understanding the polymer indeed present on the membrane surface. Okay, so I skipped this part. We have done a lot of synthesis to prove this particular concept. So I will just go to the the final last 4 minutes. 4 minutes I have? 3 minutes. Okay, 4. This clock shows 4 minutes. Okay. So the last 4 minutes, I'll just show you that what's going on in the lab in last 2 years. Okay. So now the question we ask, okay, can you build a fully biodegradable polymer? Okay. Basically, the hydrophobic chain, we build it with a simple polycaprolactam. It acting as a hydrophobic, and then this particular domain, we use again a polycaprolactam based system, so but it have a cationic charge. So in the event you expose to any biological system, the polymer is 100% will be degradable. Okay, so you don't put any waste into the domains. And also what we thought that how the topology plays a role. Suppose you make a linear diblock copolymers, or you make a star-shaped diblock copolymer, how they actually interact on the membrane surface. Okay, so that would be very exciting to look at from a polymeric perspective. And then these structures, suppose which of these structures are very smart enough in order to carry the drugs, so that you can actually, the polymer can make a pores on the bacterial membrane. At the same time, the polymer also can carry antibacterial drug, where I can have a combination therapy of more than one incidence, where I have maximum efficacy in treating the bacterial infections. Okay, so that's the question we asked, and this is where we build the structures. So we could able to build a very well defined structures where the polydispersity for the linear diblock was little bit higher. It was 2.1 the polydispersity. Still we have problem of controlling, but the molecular weight this is about 20,000. We could also build a same combination, same core, using that we can make a star block polymer, but the dispersity was 1.2. Okay, we have a good control over that, and we put this imidazolinium, this imidazole link with a long tail. We found that this particular arrangement necessary in order to maximize the bacterial impact. Okay, so we have variety of other structure we trace, but I'm just only going to this one example. Okay, so once we do that, now the question we ask, can we actually, how these molecules actually affect on the bacterial membrane? So we use this spread plate technique. Don't worry about the details, but the thing is that in the absence of the polymer treatment, you can see full of all the bacterial colonies in a control. This is your positive control. The negative control is that use ampicillin, that is your typical antibiotic. You can see there's no bacteria. Okay, almost everything is killed. These two are controlled. Now I have a star polymer as well as a linear polymer. I look at the populations. So this method we used for incubating at various time interval. So we actually looked at various time interval how fast the linear polymer incubate compared to the star polymer. It's quite surprising because star polymer is a 6 arm. You would anticipate that they should go and impact more than a single arm with a linear polymer. But that is not the way the biology functions. Okay, the biology functions in a different way. And this is a beautiful article. Okay, people who are working in this area, I would ask you to pay attention on some of these references. 2009 published by Freshe. He proposed a model how a polymer structures actually interact on the membranes. For instance, if you have a they have actually predicted for many structures, where I only summarize for the structures which are important for this particular talk. Okay, if you use a linear polymer, this is suppose any membrane, it could be a bacterial membrane, it could be a mammalian membrane or anything. Okay, so if you have a membrane, there's a resistance generated by the membrane. If you have a linear polymer can pass through this membrane with a certain energy, for instance. Suppose if you have a star-shaped polymer with identical molecular weight, suppose one of the arm of the star, suppose it penetrates through the membrane, now other arms will pull it out. Okay, so it will not allow the other polymers to go through there. So basically, each arm, suppose the amount of the energy required for star polymer to cross the membrane Suppose you have a 6 arm, it's 6 times higher than the linear polymer which can cross. Okay? So this model is very important in terms of drug delivery or people looking at in vivo analysis using a different polymer structures, and it works very well for renal filtration, how the polymer nanoparticles are filtered in your kidney. Okay? So now the similar model, in fact, it's actually applicable. Even here we shown the 2 minutes. Okay, even shown that it can be worked for a bacterium as well, and we see that the linear polymer actually, it can actually penetrate. So there is a low energy barrier for linear polymer to penetrate compared to the star-shaped polymer. As a result, you can see the linear polymer actually inhibit the bacterium much more stronger than the star-based systems. And then I'll skip this slide. Okay, just I'll go to the final one. Okay, so now from this analysis, okay, last 2 years of work, we identified the star-shaped polymer is a very unique compared to a linear dye block, where it can actually carry a significant amount of the anti-cancer drug. The anti-cancer drug which are treating with the azithromycin is a clinical drug. People use that, and this particular drug is highly water insoluble, and there are a lot of clinical limitations with that. So when we take this drug in your polymer, so this is what the free polymer does. The free polymer actually inhibits your bacterium at a very high concentration. For example, 50 microgram per ml. This is my last slide, Prasad. I'm done. Okay. And so if you take a free drug, the free drug actually inhibits the bacterium at 50 microgram per ml. Okay. So now, if you look at that, if I use a combination of the both, actually I don't need a 50 microgram, I need only 1 microgram of my drug, which can inhibit your bacterium almost 99%. So basically, you bring a polymer structures along with the appropriate clinical antibiotics which used, one can actually generate a combination therapy of disease in a bacterium disease, and that's the direction which you are moving on. So now I'll come to my final slide. Okay, so this is what I told you that initially, how much is GDP you clear, how much you do that. You must be wondering that maybe I'm talking something which is not relevant today. No, it's very much relevant today. Okay, so when more and more people now, many colleagues in many new institutes or my generation people, you see that a lot of people are working on a biomaterial science. But there's a reason for that. Maybe do we solve all the global problem, or the 20% GDP, it can be avoided by doing it, which I do not know. Okay. Okay, but maybe happen, because only 30 years down the line it will come. Probably I wouldn't be alive or not alive, okay, because 2050 the statistics. But what we generate as a young generation in this country is more important. Are we doing a stamp collection? So many people who are working drug delivery or biomaterial science, people say that, hey, they are taking something, they are putting some data they are getting, okay. That kind of impression is there in global scenario or even Indian scenario. But that is not the fact. So what we are trying to make in this country is that, or in general global, that we generate a next generation students, expose them using a smart way of using a chemistry or a polymer chemistry, any chemistry. You make a students which will be a future leaders or inventors, where the world will be in a safe hands when we are going to face any pandemic in 2050. I think that's the message I want to tell them. Some of our work which we do, probably 50% goes like a stamp collection. It does not matter. But remaining 50% even does this job. It will be a very amazing for the world population in 2050 to come. So with that, I conclude my talk saying that this work was done by Ruma Ghosh, a brilliant student, PhD student, started from the synthesis till the biology. She's executed the project entirely, and I thank you for teaching her also this entire topic to me. And these are other group members. They're presenting posters. Maybe I suggest you go through that. I thank Amrita for allowing me using her biology lab facilities. Without that, I could not able to this work, and Rupali, her student, for the help, and SCRB and ICR Pune for the funding. And finally, thank you for listening to this talk just before lunch. Thank you.
Thank you, JK, for a nice talk. Again, 2 or 3 quick questions.
Can I have water bottle? I am hydrophobic. Yeah, thank you.
Yeah, go ahead, please.
Sir, as you mentioned in the presentation that cationic polymers, whenever they come in contact, they may cause a cytotoxicity. How could you achieve the bacterial specificity for those polymers?
So see, if you have an appropriate amphiphilicity in your polymeric backbone and appropriate charge balance, okay, so you can actually have you can minimize your basically your cytotoxicity. That's what I mean to say that, okay. So but same time, when you have appropriate geometry of your polymer structures, you could able to selectively inhibit the bacterium, because bacteria, if you look at the mammalian cells, they are deuteranic in nature. On the other hand, if you look at the bacterium cells, they are actually anionic in nature. So you can have a preferential attack on the bacterium compared to the mammalian cells. Okay, so
So by controlling the charge on the
Charge
polymer.
and the structures, everything goes to the player role. That's why I showed a linear versus, if I take a star polymer, even the topology also plays a role, okay? So that's kind of information is lacking in the literature. So now not only my group, there are many groups are generating this knowledge. Probably in 5 years down the line, we have a correct answer. Yeah.
Okay, thank you, sir.
There is a question there.
Very elaborate word. Just a doubt. Bacteria, if it has cell wall, by membrane you mean the cell wall and the membrane, whereas in mammalian cells only the membrane.
Well, see, the data which I shown that is immature for me to say that only because of the membrane attack, your mammalian cells are become toxic and non-toxic. There are many other pathways it takes. Okay, but the first step, your molecule has to attack on the membranes, correct? So the membrane crossing is the most crucial part for any nanoparticle or any systems. So in order to take up by either by bacterium cells or by mammalian cells. So that barrier is playing a very crucial role.
No. Cell wall.
Correct. Cell wall has that. So for example, you have a gram-negative bacteria, you have a gram-positive bacteria, correct? The cell walls are different type, but you could able to inhibit both. Yeah, it's not specific to only gram-negative or gram-positive, but we have data only for gram-negative. But eventually, you can do it for both the systems. It's not restricted to one system. Yeah.
1 minute. There is one question, and then next to you, and yours will be last question. Yeah, please.
Hi, sir. Nice presentation. So I have one query. Did you target it to the bacteria in the biofilms or only?
No, we don't have the expertise. If we have expertise, I would love to do that actually. Yeah, we don't have the infrastructure to do that.
Yeah.
So we are just developing and testing it in a normal bacterium. Yeah.
Sure. Thank you.
Yeah, last question, please.
Yeah, very nice presentation, sir. My question is, you mentioned these are all oligomers.
Where are they?
Yeah, here.
Oh, okay, sorry.
You mentioned these are all like oligomer. It's not a polymer, right?
No, you can make 100 units of polymer, or you take 10 units of oligomers. The effect seems to be same. That's what I mean to say.
Okay.
But when you have 100 units of polymer, you can do many things. You can encapsulate drug and deliver it. But you have 10 units of oligomers, you don't have that access. Because I have to skip because of the time limit. Yeah.
Okay. What about the conversion of these monomer to polymer?
Then you can have a full control over that. Absolutely.
And my question is like a residual monomer. Is there any residual monomer present in the polymer?
No, that are not taken care of.
Okay.
Everything has been taken care of, believe me. Yeah.
Okay. Thank you.
Yeah. I think with that we will close this session. Thank you very much, both the speakers, for an excellent talk. Please wait. I think we need to at least respect the speaker. We'll give you a memento. Please wait for 2 minutes.
Okay.
Yeah, there are a couple of announcements. So we request all of you to assemble in front of the main building for the group photograph. And then after this session, there is a poster session and lunch followed. The lunch if they don't appear, we'll give the award.
A very good afternoon. Am I audible? Okay. A very good afternoon. I welcome you to the last session before we start with the music. Before we start, I have a small announcement to make. Those delegates who are not SPSI members, we request you to register yourself near to the registration desk. There is one SPSI registration desk. You can go and give your details if you want. Okay. With this, I would now like to request Director NCL, Dr. Ashish Lele, to come and chair this session.
Thank you, Samir. So we are nearing the close of the this wonderful symposium. This is the last session. There are 3 parts in this last session. The first is a panel discussion, which is on building a career in industry and a perspective. And you will hear, this is especially for students really, you will hear from some of these stalwarts who have worked in industry for a long time and have understood what R&D means to industry and how it is normally driven in industry. So that perspective for students is increasingly becoming more and more important, and we wanted to keep this panel discussion so that you can hear from the people who have done it all. So that's part 1 of this session. Part 2 of this session is 2 plenary talks from 2 very, very distinguished speakers, and I will introduce them afterwards. And the third part is the distribution of awards. So we will start with the first part of this session. This is, as I said, a panel discussion. The moderator for the panel discussion is Dr. Raman Ramachandran. He is the former head of BASF South Asia and the chairman and managing director of BASF India Limited. A very, very quick introduction to Dr. Raman. He did his PhD from the University of Adelaide in agricultural chemistry. He actually spent 1 year at NCL a long time ago. But after that, he thought that it was better to go and do a postdoc. So he went to Wisconsin, did his postdoc there, and then came back and joined industry. He joined BASF at some point of time, spent 22 long years at BASF, rose through the ranks and became the chairman managing director of BASF India Limited. Post his retirement from BASF, he is now the managing director of PI Industries and also a professor of practice at the KJ Somaiya Institute. Thank you very much, sir, for coming here. Please, sir. I'll quickly announce the names of the panelists. The first is Dr. Gopakumar Nair. He is the designated partner at Nanlex Associates LLP. Dr. Gopakumar Nair has also had very long association with NCL. He was telling me that some of his best days of life were spent at NCL. So, sir, thank you so much. And we have, of course, had long association with Nanlex. Dr. Rahul Shingte. Rahul is R&D Manager, Technology Platforms, India at Solvay Specialty Polymers. Rahul is also a product of NCL. PhD with Dr. Prakash Vadgavkar from NCL and been at Solvay for quite a long time now. So Rahul, thank you for coming. Third is Dr. Saptarshi Ray. Saptarshi is Deputy General Manager at Bharat Petroleum Corporation Limited, also an NCL product, and one of those who has, after his PhD, taken up a job in industry and been there for quite some time. Next is Dr. Anil Gaikwad, Business Head at Cosmo Films Limited. Thank you, Anilji, for coming. And finally, Dr. Shaswati Pujari, Senior Manager at Unilever. Thank you, Shaswati, for coming.
So as the panelists settle down, so thank you very much, Dr. Ashish, for that kind introduction.
And we have about 3:30 to wrap up the session.
Absolutely.
Thank you very much.
We might get carried away, so please feel free to indicate to us. So thank you very much. It's a great privilege to be back here. You know, I was here in 1987 with a Scientist B job with colleagues like Dr. Badiga, who went on to make a great career. Professor Mashalka was here at the time as head of the Chemical Engineering Division. So, sir, it's a great honor to be here with you in the audience, and also my current boss, Vice Chancellor Professor Pillai of the Swamaya Vidya Vihar, where I'm currently engaged as Professor of Practice, and to all of you. So before I get into the panel, I thought for those of you who are kind of students finishing PhDs, maybe doing your postdocs, and kind of wondering about where do you go next, I thought I'll give you a quick perspective of what the chemical industry looks like, and why R&D and innovation is extremely important and vital to the chemical industry, with a few statistics and a few kind of opening remarks, so to speak. So the global chemical industry today, actually about 2-3 years ago, was valued at $4 trillion. Yeah? Just to put this in perspective, India's GDP is $3.2 trillion. So, you know, we have a chemical industry which is much bigger than India's GDP, and mind you, India's GDP is actually the 6th biggest GDP of a country in this world. So that gives you an indication about the size of the industry that you're dealing with. Of that, what we call the specialty chemical industry is about a $1 trillion industry. And it is a specialty chemical industry which really calls for research, calls for innovation on a daily basis. So we're still talking about an industry that is a $1,000 million industry. So there is a lot of opportunities, and it is a growth industry. It is growing globally at about 3 to 4% on an annual basis. Come to India, our industry, in India, the chemical industry, about $180 billion. Of that, about 3-4 years ago, the specialty chemical industry was estimated about $32 billion. However, the most important aspect of the specialty chemical industry of India is that it is growing at about 14-15%. So by about 2024, 2025, the size of this industry is expected to double. And so you can kind of imagine the kind of huge opportunities that we are sitting on, and hence if you are a good technologist, the kind of employment opportunities that is there. The chemical industry in India is a big exporter. However, probably not very well known fact is that actually we have one of the biggest trade deficits because of chemical imports. Our trade deficit for chemical imports just last year was $15 billion. And in terms of your rank, the big contributors of trade deficit, obviously the gas and oil is number 1. Then I think the ranks are number 2, probably gold, number 3, probably today semiconductors. So we are actually in the 4th or 5th. So you can imagine from a policymaker's perspective, from a fiscal prudence perspective, this is an industry that really calls for Make in India. Okay, so that's just to give you a perspective of the size of the industry and the opportunities that are there. It is also an industry that has always called for innovation, and innovation has been a big driver going back to 150 years when the innovation, the primary driver for the innovation was scaling up. When we had chemicals that were derived from plants, for example, indigo. You know, how do you scale up indigo? Because there are a limited number of plants, and that's when chemistry came in and really helped us to scale this up. And when you talk about synthesis of ammonia, which led to the whole fertile, those all innovation was about scale up. And then there was a phase where innovation was driven by what you may call as regulations, because there were some incidents that society took notice of and regulations came in, and innovations became the primary driver. And after that, the big factor for innovation was performance. Because this is an enabling industry, it is an enabling innovation center. So that's been the big driver. And today, of course, sustainability, net zero, these are all the factors that really drive innovation. So it's an industry that really calls for research and development, it really calls for innovation. And just to put it in perspective, the industry at a global level spends anywhere from 6 to 10% on research and development. In India, right now, it is about 2 to 3%. We are still low, but if you look at what was the investment in R&D maybe 10, 15 years ago, which was probably less than 1%, I think we are also making rapid progress. And you will hear about this from some of the industry stalwarts who are here. So that's the kind of context. And with that context, I'm going to go straight into the panelists. And I'm going to request each of these panelists, and you can see here panelists who are generation A, like Dr. Nair, to generation Z or X or whatever you may call, this young lady here in the middle, Saswati. So you're going to hear a spectrum of experiences, how they. So I'm going to request each one of you, maybe with a lady.
And we are also honored by the presence of Dr. Mashelkar. Wish him well.
On behalf of the organizers of this symposium, we'd like to thank all the panelists and the moderators. So from our side, thank you so much.
Great. We've had a fantastic part one of this final session of the conference. Really wonderful to see the very positive feedback from industry. And I think the message has gone very loud and clear to the students. So we hope that NCL becomes a platform for many, many of you to become our ambassadors in industry. We will now come to the second part of this afternoon session. This session is specially organized to felicitate Dr. Shivram, who's sitting right at the corner. Shivram, will you please come here right in the front? So this celebration, of course, is on the occasion of his 75th birthday. As all of us know, he is the doyen of polymer science in India. But more than that, he's also a champion of science-led innovation and commercialization, an aspect that Premnath also touched upon in the morning session today. He's also a thought leader par excellence and also an institution builder who particularly in his tenure as the NCL director put in place several enabling R&D management systems, created new labs, conceived very ambitious mission programs, and also started with Premnath one of India's most successful incubators, as you saw in the morning session today. Dr. Shivram is also an inspiring teacher and a mentor. His special lectures on polyolefins, in which he blends beautifully science with the amazing history of polyolefins, has been some of the finest lectures that I've had the privilege to listen to many times. And presently, he teaches a very sought out undergraduate course on sustainability at ISER Pune and ISER Kolkata. Dr. Shivram is a voracious reader. He reads everything from scientific papers, patents, trade and business literature, management and policy documents, all the way to books on humanities, arts and literature. A visit to his room, you will see piles of papers and piles of books that he intends to read, and I find it amazing that he finds time to read them. He is also an ardent fan of a particular genre of music, which all of you will get a flavor of in today's afternoon's concert. Now, at his behest, we have decided to felicitate him differently by organizing two public lectures, which will be delivered again by two of India's most eminent thinkers and very, very inspirational leaders, our own Dr. Mashelkar and Dr. Ajit Ranade. The first lecture will be delivered by Dr. Mashelkar, and it is my absolute privilege and honor to introduce him formally, although I know that he needs no real introduction to this audience. Dr. Mashelkar is known for many, many things. His world-class scientific research, his transformative science and innovation leadership, his pioneering new moments such as protection of traditional knowledge,
his help in creating strong and yet balanced IPR systems, and his inclusive innovation movement based on the concepts of Gandhian engineering. And the list is very long. He has been clearly one of the most influential thought leaders in shaping science, technology, and innovation policies in the post-liberalized India. Dr. Mashelkar has a very long list of honors and awards, and if I were to read the entire list, then he will have no time to deliver his lecture. So I am going to restrict to some of the most important ones. The President of India has honored Dr. Mashelkar with 3 of the highest civilian awards, the Padma Shri in 1991, Padma Bhushan 2000, and Padma Vibhushan 2014. He has received more than 45 honorary doctorates from universities across the world. He was the first to win the TWAS Lenovo Science Prize in 2018, and the first and the only scientist so far to win the JRD Tata Corporate Leadership Award in 1998. He is only the 3rd Indian engineer to have been elected the Fellow of Royal Society since 1660, and he is also the only Indian from India to be elected as the Fellow of US National Academy of Inventors, Foreign Fellow of US National Academy of Sciences, Foreign Associate of US National Academy of Engineering, and Foreign Associate of American Academy of Arts and Sciences. Everything that the US has to offer to a research and technologist and an innovator. Dr. Mashelkar has held many key positions in his illustrious career. He was the director, of course, of this wonderful lab, NCL. He was also the Director General of CSIR. He is the chairman of the National Innovation Foundation, the president of Global Research Alliance, the president of INSA, and the president of UK Institution of Chemical Engineers, the only Indian to have done that so far. He has been a visiting professor to several universities worldwide. He is the chancellor of many universities, the chairman of Research Advisory Council of many other universities, and he is also equally sought after as a thought leader by Indian corporate world. He chairs the board of some of the largest companies, including the New Energy Council of Reliance. Dr. Mashelkar has made immense social contributions. He has chaired many CSR committees. And in 2007, he set up in his mother's name the Anjani Mashelkar Foundation, which gives the Anjani Mashelkar Inclusive Innovation Awards to recognize innovators who make high technology work for the poor. Dr. Mashelkar, we are truly grateful to you for accepting our invite to come to NCL and deliver this lecture, which is titled very rightfully, Doing and Leading Science and Innovation, Some Lessons from My Own Journey. Ladies and gentlemen, please put your hands together to welcome Dr. Mashelkar on the stage.
Just thank you, Ashish, for those wonderful words. What a fantastic feeling. Very happy to see the auditorium full. I love auditoria which are not just full, but really house full. Great feeling. I entered this laboratory on 15 November 1976, which makes it almost 46 years. I don't know how many of the students were born at the time I joined this laboratory. And whatever I am is because of NCL. Proud to stand here and speak to you. And what an occasion. Sivaram, 4 November, birthday, 75 years, many happy returns of the day. In fact, Ashish, you could have been gracious and given me 45 minutes to speak on him rather than on the topic. And I would have done that. Rajgopal, our Dr. Pillai is here, and during his felicitations, I thought I spoke about 15 minutes about you, and that was not adequate. It is there on the YouTube, but I don't have those 15 minutes. So I would simply say that Sivaram is not an individual, he is an institution. If you look at excellence and relevance, which we have been talking about, he combines it like very few that I've seen in my life. Excellence, yes, I would rate him as the finest polymer scientist that India has ever produced. And that I can say by considering all that everyone has done so far. That is his excellence. But if you look at his relevance, the way he has balanced it, can you just imagine, I don't know how many Indians have 51 US patents. It's amazing. It's an amazing record, but most importantly, I would say that there are very few scientists I've met in my life who have a deep microscopic view as well as a telescopic view of the big picture. And I witnessed it all over. When I was director of NCL, for example, there were many occasions where we had to look at the way we should move forward, like when we had 24 July 1991, liberalization, opening up, the world was going to change completely, our license to copy was going to vanish. And I remember he was instrumental in creating a blueprint about NCL into the future, post-liberalized India. When I was Director General of CSIR, I remember the large number of committees which he chaired and guided us in terms of where to take CSIR, what is the new direction that we should take. Sivaram, you had told me not to speak about yourself. And I will tell you why I'm doing that. Because you always listen to me, but once you did not listen to me. So I'm not going to listen to you now. And I'll tell you what. After I stepped down as a Director General, I wanted Sivaram to be the Director General of CSIR. I had a couple of hours of talk with him. He said, let me think about it, and I'll come back. And you know how he came back? With a 5-page letter, and he has a fantastic handwriting, by the way, and great English, so you can see the combination, on why he should not be accepting this particular offer. Sivaram, I will never forgive you. You did not then listen to me, and therefore I had to disobey you and say a few words about you. God bless you. You and Rama make a fantastic pair. You have been part of our family. You are in our hearts all the time. 75 years is a number. I like the number 100. All right? So all our best wishes, choices, blessings on behalf of me, Vaishali, all Mashelkars, all NCLites, all family members of CSR for your great journey towards your century. Thank you. Now I feel happy. So let me take you through what I have said in the title, Doing and Leading Science and Innovation, some lessons from my own journey. I decided to talk about my own book of life, rather than giving you a sermon by reading some other books. And my book of life is all here. NCL, CSIR. That's the only job I have done, by the way, in my life. There is no other job. Okay? So what did I learn? Not that everywhere I succeeded, I failed also. What are the lessons that one learned? So there are two parts. The first is doing science, and second is leading science. As far as doing science is concerned, we talk about Indian science. And to me, what is very important in science is that you can't have sea of knowledge where you keep on putting buckets of water which make no difference. You have to create waves. You can't let somebody open up new windows and then all of us look through those windows and work. Bednorz-Muller create a breakthrough on superconductivity, all of us rush and do that. All right? Graphene, breakthrough, everybody works on graphene. Carbon nanotubes, breakthrough, everybody works on that. No. We have to create our own windows. I remember one fellow of our society had visited, and I said to him, how do you elect FRS? FRS is an honor, many consider very high honor after Nobel Prize. You know, in fact, I will narrate a little story about. And then he said, how we select it. He said, supposing you have written 576 papers, and your name is XYZ, say R.A. Mashelkar. Supposing we put a delete button on that, that means you were not born at all. Would science have taken a little left turn or right turn? That's the kind of science we should be doing. All right? And aspiration of Indian science has to be that. I'm very proud to say that National Chemical Laboratory has done that. And the evidence of that is here. I remember getting elected to Fellowship of Royal Society 1998, entirely on the basis of the work done in this wonderful laboratory. You can see me shaking hands with the President of Royal Society, Aaron Klug, a Nobel laureate, but also signing in the book where Newton has signed on page number 9. You know, from entire CSIR, there are only 2 in 80 years who have got this honor, myself and A.P. Mitra. I'm not happy. I want not 2, but 20. All right? How can we raise our level of science to that? The next honor, which is considered most important, is US National Academy of Science. In fact, Sir Richard, the Nobel laureate, was speaking there on the other day. During dinner, he was telling me he's not elected to US National Academy of Science. He should have been elected long time ago. In fact, Sir Harry Kroto became Nobel laureate first, and then he became US National Academy of Science. I'm one of the 2 engineering scientists elected in the last 248 years. Again, work done in this particular laboratory. I think that's the sense of pride we should be sort of carrying on, and that's the level of science we should do. So in order to do that, what are the 4 lessons that one learned? First, is nothing like basic science and applied science, only science and its application. Many times, you know, Narsab, we have this debate, scientific research, industrial research, this, that, and the other, etc. No, that was my first lesson. Second, only those who say the first word or last word in science are remembered, so be first, not even second. Third, be bold. Chase anomalies. They will open up windows to new knowledge. And fourth is borderless science. They say parachute works best when it is open. Mind is also like that. It works best when it is open, borderless. And I'll give you some examples of that. Now, if you see this great laboratory as we enter, and I entered it on 15 November 1976, what struck me was this fantastic statement. The purpose of this laboratory is to advance knowledge and to apply chemical science for the good of the people. I have not seen a more profound statement. You know, it's incredible. I remember when I joined on 15 November 1976, I was asked to set up a polymer science and engineering division. None existed. We had a good polymer chemistry division, but not polymer science and engineering. Dr. B.D. Tilak was here, and I remember those days. I was trained in rheology and non-Newtonian fluid mechanics, and I needed an equipment. It was an experimental science, so I needed an equipment. And what was that equipment? Weisenberg rheogoniometer, model R18, I still remember. It would take 2 years to get it. And particularly for young students, you must understand under what circumstances we did research at that time. Why would it take 2 years? DGTD clearance, not manufactured in India, a poor country, not $2,000 GDP per capita, $200 GDP per capita. All right? So what do you do? And that's why I always tell students, among my 5 Machelkar mantras, one of them is, you keep on knocking on the doors, they don't open, open your new door yourself. So that's what I did. I said, I'm not getting this equipment. What is the equipment God has given me? This. So I went into modeling and simulation. All right? And leveraged, because we have been talking about scientific research, industry research here. I was a consultant to Indian Organic Chemicals, which was manufacturing polyethylene terephthalate, polyester fiber, basically. 6,000 tons per year, Sivaram at that time. Reliance makes 6,000 tons per day now. That's a different. But their technology was an imported German technology. They did not know what was happening in the reactor. So I had a dealing with them, and I said, one of my PhD students will work on it and will tell you what is happening there. And then emerged series of papers. My student's name is Ravindranath, and he and I wrote these papers, series of papers, end to end. Trans-esterification, ester interchange, pre-polymerization, polycondensation, up to spinning, whole range of things. And I'm very proud to say that in that impoverished laboratory, at that time we were able to create breakthroughs. Like one of the last papers that we have in AISH journal, it went to Bob Secor, who was the Chief Technology Officer of DuPont, who were leaders in polyester. And we had solved some anomalies that appear in polycondensation reactors. And he told Bob Reed, the AISH journal editor, that for donkey's years we have been working in this. This is the first time we come to know what's happening there. And Ravindranath became, by the way, later, the Chief Technology Officer in the specific field of polymer for AspenTech, which is the greatest modeling and simulation company. We could do that because it was the power of ideas that was actually used. I want to say this because this was done in an impoverished laboratory. And you know, 77 to 82, that was the time of work I did. I got this SS Bhatnagar Prize for those 5 years. Now you would say, was it industrial research or was it scientific research? If it is industrial research, how did Ravindranath get his PhD? How did I get Bhatnagar Prize, which is supposed to be for science? If it's scientific research, how did Indian Organic Chemicals transformed their industry, basically in terms of productivity, product quality, new products, etc. So forget about this distinction. I think that's the first point I'm making. And while on the subject of the Bhatnagar Prize, Ajit, come here. No, please come, please come. Give a big hand to Dr. Ajit Ranade. Yeah, you can shift there so that you don't have to remain tilted for... why don't you shift there? You are sure? Yeah. Okay. Yeah. But you are tilting to your left, remember that. Okay. So, talking about that, the rich tradition of NCL, I want to make you feel proud. Ashish Lele here receives Bhatnagar Prize at the hands of Dr. Manmohan Singh, the Prime Minister, by the way. And Ashish, you can see my hand around you, which has been there from the time you met me in Delaware for the first time. And that's a fantastic tradition that we have. Prime Minister giving the awards. 1958, the Bhatnagar Prize is started. This year, we are very proud that Amol Kulkarni got the Bhatnagar Prize. Amol, where are you? Are you here in the audience? He's missing. Huh? All right, okay. So this is a matter of great pride. He received it from a Nobel Prize winner, not from our Prime Minister. The tradition has been broken for the first time. The Bhatnagar Award arrived in post, I understand. So I want to congratulate you for sending the right message to those who matter by giving the award at the hands of the Nobel Prize winner right here in NCL. Very important. That's the first lesson. The second lesson is only those who say the first word or last word in science are remembered. So be first and not second. Now here, I remember receiving this Lenovo Science Prize. This is the highest science prize of the World Academy of Science. And the reason I received it was some contributions we had made where we had done many things for the first time. The word first is very important, by the way. You'll keep on hearing the word first. Leadership is being first, not being among the first. So, it was Ashish, Shiny, and many of our young colleagues where we had a wonderful group on stimuli-responsive gels. We were the first ones to discover self-healing, first ones to discover self-organization, first one to do the discovery of biomimicking switching hydrogels with enzyme-like activity, first molecular level mechanistic interpretation of stimuli response, your lattice fluid hydrogen bonding models, you remember? First creation of a magic molecule called A6-ACA, which has found applications amazing, ranging from elastic entry implants to biomineralization to stem cells to well drilling. The last point is very important. Normally, others find a material and then we are expert in finding applications. Here, we found the material and the rest of the world, including MIT, is busy finding applications. That is the reversal. That's what we want. That is leadership. And just very shortly, the biomimicking self-organization, the chiral 6-aminocapric acid gel, as you can see, could do the magic of a cylinder turning into a hollow coconut-like structure and then reversing it. Nature had called it Indian rope trick, if you remember. And this has great significance in terms of the way it organizes. If I was giving a total scientific lecture, I would have explained to you how it was. And then we were the first ones to find self-healing biomimetic smart gels. You can see here A6-ACA hydrogels. There are two pieces which are put together, and when they are brought together, how rapidly they heal. This was considered a kind of a breakthrough for a variety of reasons. It was a paper that appeared in Proceedings of National Academy of Science and was highlighted by them. The issue was about the tightrope trapeze artistry, you know, balancing the hydrophilicity and hydrophobicity. And the magic of this particular molecule, that series that we had created, the copolymers, was that if you had, for example, if you look at the methyl groups, healing would take place most strongly when the methyl groups in the side chain were 5. Below that, they wouldn't. After that, they wouldn't. And that was a sort of a magical molecule. Again, there are reasons why that happens. And then there were a large number of applications that actually came up. For example, this stem cell engineering, promoting stem cell adhesion, shape, migration, and differentiation. And what was found, this was the work done in University of California, San Diego, the stress adherent spread best at A6-SCA compared to matrices, which containing fewer or higher methylene groups in the side chains. And you can see that impact in terms of the adherence as seen being the highest here. Then the superior matrices for osteogenetic differentiation. This hydrogel biomineralization could be enhanced very significantly by using that. Then amazing application, we would have never thought about it. You know, when you have these circulating materials for drilling or fracturing, there is a loss of circulation fluids in drilling wells. And the crux of this particular invention was that introduce this hydrogel based on our material. Upon lowering pH, there is instant agglomeration, and when the pH goes higher, it goes the other way around. And you can see the trick, as a matter of fact. At high pH, it is like this, low pH, it is like this. Again, it is sort of reversible. This was a Halliburton patent. And this was amazing. This was work done by none other than Bob Langer in MIT, creating the elastic enteric implants. And their major problem was the gastric retentive devices must have facile delivery, prolonged gastric retention, complete safety profile, and the currently existing enteric polymers were rigid and brittle, and there was no device for combining enteric plus elastic properties. And where did they turn to answer? They created a pH-responsive supramolecular gel, which are stable in acidic conditions as well as in alkaline conditions. And this produced the winning candidate. In fact, very proudly, I feel like hanging this on the wall. MIT recognized the magic of A6-SCA by making this statement in their Nature paper. Although there have been extensive studies and commercial examples of gastric retentive devices, science is concerned, be bold. Chase anomalies. We'll open windows of new knowledge. And there, I must say, I have a lot of fascination for this from old days. In fact, I have a paper in Current Science, Fun and Joy of Science, learning from anomalies and discontinuities. And my early fascination, I remember, there was this anomalous separation in viscoelastic fluids, anomalous transport phenomena in rapid external flows, anomalous convective diffusion in films of polymeric solutions, unusual retardation and enhancement. You will see the words, anomalous, 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 and unusual. Go for the unusual, not for the obvious. A data point which falls outside the prediction has a message for you, has a new science for you. Please keep on looking at it. Normally we discard it as being an experimental error. Don't do that. I chased it all along, and there is a benefit. Like in convective heat transfer, heat flux increases with increasing velocity, right? But in rapid viscoelastic flows, there was an anomaly that heat flux will not increase, but it will remain constant despite increase of velocity. And I remember working with Marucci and this particular paper, we looked at the leading edge and say it is not the inertial and viscous forces which balance, inertial and elastic. And that is independent of the velocity, all right? Square root nu lambda, nu being the kinematic viscosity, lambda being the relaxation time, which remains constant. That was a breakthrough. But for that, what did we have to do? Prandtl analysis was considered standard from 1937. We had to have irreverence for that Prandtl analysis. It can't be used here, and create a new analysis. You know, as one Nobel laureate has said, our challenge is not in creating new ideas, escaping old ideas and creating new ideas. That is the kind of spirit we must have in our research. There was this norm right here in this laboratory. You know, you take a cube of sugar, put it in water, it will take a long time. But you create a microfine sugar, instantly dissolves, right? So you keep on reducing the size and dissolves faster. With polymer particles, the issue was, it comes to a particular size, and after that, no matter how fine you go, it doesn't change. And that had remained unresolved for many years. And in this laboratory, a PhD student, Devota and Ambeskar, they worked together and showed that when a solid polymer particle dissolution time remains constant, we had a paper which showed, first time we showed the critical role of reptation dynamics the resolution time becoming equal to repetition time below critical size, because as you know, molten polymers are like entangled chains, right, moving in a snake-like motion, and using that analogy, finally, that time for reptate became the critical part. So there was completely new sort of science that came out. So these are my 3 lessons. My last lessons that I learned was borderless science. Great breakthroughs at interfaces, not silos. Inorganic chemistry, organic chemistry, physical chemistry, polymer chemistry, biochemistry, none of you talking to each other. No. Breakthroughs will take place there. And I remember my fascination, again, when I was director here, I was invited to give the Dankwards Memorial Lecture, and the title was Seamless Chemical Engineering Science, the emerging paradigm. And in fact, we have a paper in PNAS, because after you might wonder what did I do after 2006, after I retired from CSIR. And of course, I was here as a Bhatnagar Fellow for some years, but a lot of time I did research outside. I was visiting professor at Harvard and at Sir Louis Matheson District Professor, etc. And one of that was this, as a matter of fact. And you can see something remarkable here. This is a reporter nanoparticle that monitors its anti-cancer efficacy in real time, by the way, how it is working. And this breakthrough, actually, if you can read here, here we describe biology-inspired engineering of a simple 2-in-1 reporter nanoparticle that not only delivers a cytotoxic or an immunotherapy payload to the tumor, but also reports back the efficiency in real time. And the reporter nanoparticles are engineered from a novel 2-stage stimuli-responsive polymeric material with an optimal ratio of enzyme-cleavable drug or immunotherapy effector elements and a drug function activable reporter element. But this, if you just see the authorship, it is so many multiple disciplines who have actually come together. And breakthroughs, future breakthroughs will take place here. Now I move from science. I will talk to you about science and how to do cutting-edge science and what kind of mental attitude you require, which will facilitate that. Now I move to the second part of my talk, that is leading science. And as you said, Ashish, I've been director of NCL, then DG of CSIR, president of INSA, chairman of National Innovation Foundation, president of GRA. GRA is Global Research Alliance. Just like we have CSIR India, CSIR Australia, CSIR South Africa, Fraunhofer Gesellschaft Germany, VTT Finland, DTI Denmark, TNO Netherlands, Battelle USA, etc. I was president for 10 years. Then president of IChemE UK, as you mentioned, chairman of several corporate innovation councils like Reliance and so on. Then I was senior World Bank consultant on institution restructuring. Then on several global research and innovation advisory board, jury on major global awards like Queen Elizabeth Prize of Engineering, a £1 million prize which is considered as a Nobel Prize for Engineering, and of course 16 Mashalkar committees, because as somebody said, whenever the government was in difficulty, they would form Mashalkar committee. My first committee was Regional Engineering Colleges to NIT. I still remember 1998. Why I'm mentioning this is not to show you what I have done, but I have had the fortune of looking through thousands of windows around the world. I consider myself very lucky. So my lessons that I'm going to talk about have come not just being director of NCL or DG of CSIR, but this wide spectrum. I consider myself very, very lucky that I had. CSIR, of course, I'm very proud that Jayan Narlikar in his book, Top 10 Achievements of 20th Century, he starts with Ramanujan, Meghnad Saha, S.N. Bose, C.V. Raman, etc., Green Revolution, and finally, the CSIR transformation that we all did together in 90s is considered as the top 10 achievements of Indian science and technology in the 20th century, and we should be proud about that. Now, you can see this Global Research Alliance, for example, we had all these institutions come together. And I was also fortunate as a senior World Bank consultant in reforming, restructuring these institutions from Indonesia to Croatia to Serbia to Vietnam and so on. So there's a vast experience. In all that, what did I learn? Just 4 lessons. I've learned many, but I will highlight 4, and some of which have their origin right here in this laboratory. The first is, it is the power of idea that matters and not the power of budget. extremely important to remember whenever we talk about R&D budgets. It is the power of ideas. And I remember a day here, you know, this particular photograph has been taken on 1st June 1989. Sivaram, you were in the audience. Ashish, you were in the audience. This auditorium was not just full, it was overfull. There are 1,000+ people in this auditorium, some of them sitting like this, some of them sitting like this, and so on. And at that time, we created a new vision, a new dream for NCL. Why? 1989 was 2 years before India opened up, liberalization opening up. What were we doing? Reverse engineering, copying. All right? We said nothing. Anytime our fantastic minds here created something new, which was ahead of the rest of the world. If I went to industry, they would say, but has Japan done it? Has US done it? Has Europe done it? If they have not done it, how can you do it? That confidence was not there. So on that day, we simply say, National Chemical Laboratory will be International Chemical Laboratory. What am I selling? Knowledge. What is my market? Not Pune, not Maharashtra, not India, the whole world. And then we changed the paradigm. We said it is not the power of the budget, but the power of idea that matters. And the first thing was not publish or perish, but patent, publish and prosper. And I remember making a bold statement on that day. I remember I was reading something that GE had done, and I said we should be able to license our patents to GE. And this was very bold. You know why? Because till 1989, after we were born, there was not a single US patent that we had filed. We talk about 51 of his patents. And then I'm promising that we'll export our knowledge and license our US patents. That requires guts. Where did those guts come from? They came from the Sivarams of this world, Paul Rattoswamis of this world. I had faith in them, confidence in them. I always say talent, technology, and trust, that they will deliver it. And they did. And just see what, this is just what Sivaram has done. NCL's 3 US patents on polycarbonate were licensed to GE in 1994 around $1 million. First ever reverse transfer. Strategic alliance of NCL over 10 years led to $15 million license fee. Other partnerships opened up, DuPont, Dow, Procter & Gamble. And Jack Welch, when he came to know about it, he said, if they are so good, why are we not there? And the Jack Welch R&D Center came up. And I'm very happy to say that that was the beginning of India becoming a global R&D platform. Today we talk about 1150 to 1200 multinational R&D centers here. And I had to mention that when I was inaugurating the BASF R&D laboratory, you remember that? That was the confidence we had. So in 1995, Ajit, I gave a talk when Dr. Manmohan Singh chaired it. He was the finance minister. And I say India's emergence. And as you know, I have this reputation of being a dangerous optimist. So nobody took me seriously. I'm very happy to say that in 2016, I was able to write a paper on how we had become the R&D center. All right? Some of the companies actually producing 1/3 of their IP based on the IQ of young people. And it was not just that this was being done, it was bringing a benefit of actually drawing young people from abroad. And when they would join the Gs of this world, they will join our laboratories. So it was brain drain to brain gain to brain circulation, by the way. You know, it was an unbelievable phenomenon. And Sivaram, your contributions on this are absolutely massive. We are grateful to you for what you did by demonstrating that this can be done. The second lesson is raise the aspiration. We have been followers, then we became fast followers. We would take pride in the fact that any generic, any drug that is invented anywhere, we will copy it in 18 months. Fast followers. Then we say, no, we must lead. And then we say, we must leapfrog. And then I say, no, not leapfrog. We must pole vault. Now, what is the genesis behind the pole vault? You know, I've been the chairman of Reliance Innovation Council, which had Novolaric, Joe Maryland, Novolite, Bob Grubbs, George Whitesides, whom you know as a genius, C.K. Prahlad and all that. And we created this Reliance Innovation Leadership Center here. And as you can see, the paradigm shift was growth is life. No, we said innovation as a way of life. So it has to be innovation-led growth. And in that innovation-led growth, it is not linear growth, exponential growth. And if it has to be exponential growth, then it has to be disruptive innovation, not just incremental innovation. So you have to change gears in order to do that. And I remember Mukesh said, Doc, we must leapfrog. I don't remember the context. And I said, Mukesh, do you know why the frog leaps? Because he's afraid of his predators. So he jumps a few feet, and again a few feet. Should we be doing that, or should we pole vault? The size of the pole determines the size of your aspiration, right? And then we created this program called Beyonders, leaders who were capable of thinking beyond the realms of possibility. And this book, Leapfrogging to Pole Vaulting, came as a result of that. And Ajit is here. You remember this moment when you gave us the best Business Book of the Year award. You're one of the juries. It was not bad for a scientist. Of course, I had Ravi Pandit as my co-author to write the best Business Book of the Year award. But did we do the pole vaulting? And you can see the pole vault here, how once Geo came, we became actually from 156 position, we did not leapfrog to 100. We pole vaulted to number 1, and we have remained number 1 so far, all right? That is the aspirational part. And it was not only that. When you look at 50 million users, telephone took 50 years, mobile took 12 years, YouTube took 4 years, Facebook took 3 years, Twitter 2 years, and Reliance Jio did it in 83 days. So speed, scale, and sustainability is a characteristic of pole vaulting. And I'm very happy to say, in general terms, and that is where our science, technology, innovation has to gear up to help India pole vault. We are doing that. World's fastest and largest financial inclusion through JAM, Jan Dhan Yojana, Aadhaar, and mobile. World's fastest and largest jump in mobile data consumption I showed to you. World's fastest and largest digital payments, UPI. We are way ahead of China, by the way, way ahead of the rest of the world. World's fastest and largest deployment of LEDs, light-emitting diodes. Like somebody was saying, that it is not the R&D, research and innovation, but marketing and everybody. One of you said that. You are absolutely right. Here, India moved from 0.2% market share of LEDs in lighting to 88% in 7 years, which was a record. How did they do that? They did that by innovation, demand aggregation, creating delivery systems, innovations, and so on and so forth. So you can see the pole vaulting now from starting up nation to a leading startup nation. You see, for example, this news of India produced more unicorn startups in 2021 than all previous years. That's a big pole vault. In fact, until 2016-17, approximately 1 unicorn was being added every year. In 2021, it added 42 unicorns. Not 52, but 42. Not once, 1 year, one in 1 year, 1 in 1 week, year to week. All right? Don't you see the sense of pride? I don't hear any applause, no sense of pride. What is this? We are the only country who has achieved that. I think there should be a sense of pride, and you should be able to express that. Not for me, but for the startups who have done it. All right? Am I right? Yeah. Then, most importantly for you, and I need an applause here, on demand. I did an analysis of among the startups, how many of them came from IITs and IIMs? Do you know the good news? 50% came from there, but 50% came from Tier 3 cities, Tier 2 cities, school dropouts. This is democratization of innovation. Can you just imagine somebody from Satara, Sangli, Khed, Shivapur, at the age of 26 becoming a unicorn In case you don't know unicorn is a $1 billion market cap. Can you just imagine a farmer's son rising to that? That is the aspirational India that we are actually talking about. But then, when we were writing the book, somebody asked a question, how do you assure an assured success in a pole vault? How do you make sure that when you do pole vault, you don't break your back? And that's why if you look at the book, there is this assured innovation framework. Affordable, scalable, sustainable, universal, resilient, excellence in terms of technology, and distinctive, not me too. All right? Now, this particular framework I started using when the Prime Minister talked about Swachh Bharat. There was a committee that was created under my chairmanship to select the best drinking water and sanitation technology from around the world, because we have to have the best. And we introduced that. And this framework is not qualitative, it is quantitative. You start putting numbers, basically, all right? And you could see that if you the impact was sort of enormous in terms of the selection. And you could see, as a matter of fact, in the book, a table which shows the number of companies and this matrix. And companies which were successful at one time, had 60 million customers, 80 million customers, don't exist today. Like BlackBerry, right? It had 80 million customers, doesn't exist today. Because you have to be assured every day. When they came in, they were distinctive, they were different. But they stuck to a machine for electronic mails, emails with keyboard. So when touch came, Android came, etc., they didn't change and they don't exist. So the message is that you can't be just assured one day, you have to be assured every day. Okay? And here is a case, for example, if you look at Jio, was it affordable? Yes. 4 rupees per GB and voice free. Was it scalable? Yes. 500 million customers. Was it sustainable? Yes. It is making profits. Was it universal? Yes. Look at these two ladies. Both of them are using it. User-friendly. Was it rapid? Yes. You saw 83 days, 50 million customers. Was it excellence? Yes. 4G LTE was given when the country was struggling with 2G and 3G. The West. And was it distinctive? Yes, in many, many ways. So that's what we need. And best of companies might mess up, like this Google, for example. I'm just showing an example because Google is the ultimate in innovation. They created this Google Glass, which was distinctive, which was excellent, but it did not meet the other criteria. For example, user-friendliness. When you're using a Google Glass, one did not know whether they are downloading your conversation, or they did not know when you entered a theater whether you are downloading a movie and so on. It vanished. So this assured framework, I'm very happy to say, has gone across diverse disciplines. Research institutes are using it. Some of our CSIR laboratories are using it. Startups are using it for spot analysis, like some of them in Venture Center, but also GeoGenNext. SMEs are using it, corporates are using it. TCS, right here in Pune, is using it. Innovation accelerators are using it. Some of the government agencies are using it. And now even there is an award called BMA, Bombay Management Association, Assured Enterprise Award. You know, the first one went to Baiju and so on and so forth. So I think what I'm emphasizing here, Ashish, is that this particular framework, it is not your TRL, technology readiness level. TRL tells you whether you are ready for current business. Assured tells you whether you will be ready for business 5 years from now. And you're once assured, you are not always assured. Every day you have to be assured. So I thought I will leave that message. And taking the analogy forward, pole vaulting, because I want India to be a pole vaulting nation. Not crawling, not walking, not leapfrogging, pole vaulting. But remember the pole vault analogy. When you are running, you have to accelerate, right? You start from zero velocity and then you accelerate. Then you generate kinetic energy which takes you over the bar. Now supposing in between you have roadblocks or speed breakers, what happens? You stop. Now that is my big concern of Indian science today. What are the common speed breakers reducing the kinetic energy? And I'm going to be a little frank here. First is bureaucracy. I remember 3 prime ministers in science congresses saying they will eliminate bureaucracy. One was when I was the president of Indian Science Congress, Atalji mentioned that. Second was Dr. Manmohan Singh in 2011, Science Congress. Third was our Honorable Prime Minister now in Mumbai mentioned that. In fact, I specifically remember they should not be solving the puzzles of bureaucracy. They should be dealing with puzzles of science, something of that kind. So there were serious intentions, but I'm not happy at all that the bureaucracy has just gone up. Second, trust deficit. Most important. You know how I was appointed? Dr. Naidumma met me in Savoy Hotel. Dr. Tilak was then the director, sent me a telex message, go and see Dr. Naidumma. He did not tell me why I should meet him, but he was my guru and you listen to guru, I went. We had just 45-minute conversation. I showed to him what I was doing. He was very impressed and he offered me job on the spot. No formal interview. And Dr. Tilak later on showed to me a piece of paper on which NCL had sent him some information about me. He had simply said, he's fantastic, grab him. That was my appointment letter. This used to happen. This used to happen in 1976. Sivaram, can we do it today? What Naidumma could do to a Mashelkar could not do to another Mashelkar during CSIR. And now I understand your ad hoc and fast, etc., it's all closed. Bring that trust back. You talk about startups. I remember when I was Director General of CSIR, 1995, in the very first meeting, I had created this science entrepreneur scheme, where you could take knowledge from a laboratory, create an industry, doesn't work, come back, your job will be intact. That was the scheme. Today, I don't have to explain to you what has happened. This is not done. You can't pole vault. Centralization, in fact, over-centralization. I will not elaborate on that. Restrictive regulations. Regulations stop. BT Brinjal, we could not. There was a regulation. Now BT mustard, yes, because regulation has changed. So you can have all the technologies in the world, but you can't do that if there are restrictive regulations. Disincentivization. I was not at all happy when I heard that all agencies are supposed to have just one award. Just one award. How can that be? What happens to the young scientist? Our Amol Kulkarni got in 2020 Bhatnagar Prize, but in 2011 Young Scientist Award. He won't be able to get that now. Because as young scientists grow, they must have that, no? This is not done. If knowledge converts wealth, part of that wealth must go to the scientists who create it. I think that has come under hammer now. What is this? What is this, Sivaram, what we did in 1995 to 2006? I think reinventing CSIR is like rewinding CSIR. If you ask me, we have to get back to those days. And static R&D investment. What was our R&D budget for the nation, I mean, science and technology budget? 0.68% in 1998. What is it today? 0.68%. China has gone up by a factor of 3 in terms of percentage GDP, and they are richer by 5 times, so 15 times. How can we compete? I'm mentioning this specifically because as I'm turning 80, if I don't show you my anguish, I don't share it with the family, I would be doing something wrong. All right? So we have to create that pole vaulting nation by removing all these red blocks. Next 2 points and I will close, Ashish. One is master inclusive innovation, make high technology work for our poor, creating access equality despite income inequality. It is extremely important. That access is extremely important. All right.
And I remember proposing this Gandhian engineering. Mahatma Gandhi had said, earth provides enough to satisfy everyone's need, but not for everyone's greed. That means getting more from less. Preserve your current exhaustible resources. The second was, I would prize every invention of science made for the benefit for all. That we do it for more and more people. So it became more from less for more. And this was when I was honored as an Australian Academy Fellow. You can see Robin Batterham, the president there. This was done in 2008. And Gandhian engineering, therefore, was getting more performance from less resource, from more people. MLM. It became famous as MLM. And then within 2 years, C.K. Prahlad and I wrote this paper in Harvard Business Review. And this is now ranked among the top 10 must-read papers in innovation. Innovation's holy grail, more from less for more. And World Economic Forum, can you believe it, within 6 months, had a special session on more from less for more. What are our biggest challenges today? The first is increasing inequality dramatically. Poor have become poorer, particularly after COVID. Hundreds of millions have gone below the poverty line, all right? Increasing inequality. And therefore, to get access through these Gandhian principles becomes very important. And I remember spreading this message around the world. As you can see, I began in Canberra, spoke in Melbourne, then Atlanta, Boston, Washington World Bank, I remember, they created Vietnam Inclusive Innovation Project of $55 million based on the principles. Bangkok, Kuala Lumpur, Manila, I was invited for a special honor by the Asian Development Bank. They incorporated it in their strategies. Here, Brussels, Cambridge, London, Munich, Paris. Brussels, European Commission had invited me to speak, and they had that Horizon 2020 program. They included that. And you know why, Ashish? Because they see you are talking about affordable excellence. Excellence, you also create, we also create, but if you make it affordable, you will beat us. So we must incorporate that. Can you just imagine what it meant? Went to Beijing, went to all these other cities. Tokyo, sustainable development goals, how to achieve them, and they found more from less for more is a key, so they had invited me for that. That became a strong recommendation, Colombo, Cape Town, Medellin, so on and so forth. Then you will say, you are spreading the message, what were you doing yourself? Well, actually, if you see, CSIR has been doing Gandhian engineering. The cheapest generic drugs in the world for the poorest of the poor, who would have created them but for us? But I did an initiative of my own, in the name of my mother, Anjani Mashelkar Inclusive Innovation Award, and that was given for Gandhian engineering, getting more from less for more. But I went one step forward. I'm not a great believer in best practice, because when you follow best practice, you are following somebody else. I'm a believer in next practice. Your next practice has to become best practice for sort of others. So this award is being given only to the next practice, who go ahead of the rest of the world. And we had this Rahul Rastogi, for example, for Sanket, the ECG device. And you can see he created monitor. This was the best practice. You go lie down and so on and so forth. No. He created a device which looks like a small mobile phone, for example. And you just take it, put your 2 thumbs for 15 seconds. Then the sensor around your heart, 15 seconds, 15 seconds. And within 5 rupees, you get an accurate ECG, by the way. This is approved by US FDA and everyone. And you can quite clearly see this was his first device, and this is the second device he created. 2 lakh devices have been sold in 8 countries now, just 5 rupees per scan. Not best practice, but next practice. You look at Mission Ingawale, hemoglobin, for example, if you want to know, what is the best practice and next practice? Best is invasive with needles and you take blood. Non-invasive, no needles. Cost per test, 150 rupees. Cost per test is just 10 rupees. And this was the first technology he created. High technology, photoplethysmography, spectrophotometry, etc., advanced photoscattering. Remember, Making high technology work for the rich, very easy. Making low technology work for the poor, very easy. Making high technology work for the poor, very difficult. And that's what he has done. And then, this was the best practice. He created the next practice, Touch HB. And what is this? This captures, all that you have to do is to look into the camera. And this captures the picture of conjunctiva and uses the method of reflectance photometry to estimate hemoglobin content. Can you imagine now in a rural village? All that you have to do is to have some cameras, and all that village lady has to do is to just go and look into it. Transformational. And our young startups are doing that. That is very important. So that is that. And finally, we have to come to taking risks, by the way. And I have done those experiments. Some of you will remember we created Kite Flying Fund, right? Chance of success 1 in 100. You remember in this very auditorium there used to be a competition and people used to very proudly say, I got the award. When I went to CSIR, I called it a New Idea Fund because Parliament was just 1 kilometer away. They would say public funds are being used for kite flying. So I called it New Idea Fund, and there were breakthroughs that came up, like spin computing and so on and so forth. And then we created this NIMITLI, New Millennium Indian Technology Leadership Initiative. And that was based on talent, technology, and trust. It became India's biggest ever public-private partnership, grand challenges, seeking game-changing solutions, bold financial instruments, money being given, Ajit, to private sector by the government, provided they look at some grand challenges. I had said 0% interest. In a cabinet meeting, I was not there, so it became 3%. But still, the interest rates were 9, 10%, so it was still low, all right? And risk was the key. And what was the major change? The major change is here. The major change is here because if you see the positioning of NIMITLI, you know, market certainty very high, technology certainty very high, and we had always worked in this. Markets were there, technology could be copied, IP laws, comfortable, assured market, safe entry. We said no, we shifted to this zone where markets were uncertain and technology was uncertain. And large number of projects actually came out. Not everything worked, by the way. There were many failures. But for the first time, we were taking risks, basically. And the risk benefit you can see, for example, we started the hydrogen program when no one talked about it, 2002. Had you heard about hydrogen program in 20 years ago? We started that. Thanks to you, again, Ashish. All right? And we progressed. I don't want to go through that. But today, when we see a photograph like this, which we display proudly all around the world, I display it. Ashish is here and our KPIT technologies, Ravi Pandit is here, who took the risks. And you can see this bus. This is this NIMITLI partnership. And fuel cell technology would cost around $400 per kilowatt compared to $1,000. Affordable excellence. Remember, the theme for us has to be 10x. 10 times better and 10 times cheaper. I will allow 9 times better, 9 times cheaper. But that's the way we have to think about affordable excellence. That is our competitive advantage. I want to close by saying that pole vaulting is a spirit. Bold, setting benchmarks, raising them, going over, and so on and so forth. I'm very happy when General Bipin Rawat, unfortunately, he is no more. When he read the book, he gave an interview and he said, time has come to move from leapfrogging to pole vaulting. I think a time has come to do that. And I would say, I would not talk about India at 100, by the way. I would talk about Bharat at 100. It is very important. All right? That's the true nation for us. We have to change what we do, not just renovation, painting a bit, creating revolutions, new to the world, not just to company or country, unprecedented cost performance features, not just marginal cost, entirely new products and services, not just tinkering with the existing ones, and finally, next practice, not just best practice. I'd like to end by what I said in 1998, when I got the JRD Tata Corporate Award that you talked about. I had said then, finally, 1999 should be the year where we should launch a powerful national innovation movement to propel us into the next millennium. The I in India should not stand for imitation and inhibition. It must stand for innovation. The I in IIT must stand for innovation. The I in industry, the I in CSR must stand for innovation. The I in every single individual Indian must stand for innovation. And then I had ended this speech by saying, and this was my dream, and that dream still continues, that it is only this innovative India that will signal to the rest of the world that we are not a hesitant nation, unsure of our place in the new global order, but a confident one that is raring to go and be a leader in the Committee of Nations. Thank you. Thank you. Thank you, thank you. Thank you so much. I did not realize that all your claps in between you will collect into the final one. Thank you.
On behalf of the organizing committee and all of us here, it's an absolute privilege to give a small memento to Dr. Mashelkar for his incredible speech. I said earlier that when I was introducing Dr. Mashelkar that he is truly an inspirational leader, and I'm sure all of you have witnessed that today. We have heard him many times, but there are many young people here for whom such speeches are moments in your lifetime that you will remember, and I'm sure this is going to be one of them. Our next speaker for this session is Professor Ajit Ranade, and I have the privilege to welcome him for this session. Dr. Ajit Ranade is the Vice Chancellor of the Gokhale Institute of Politics and Economics, the oldest research and training institute in economics in India, and an institute dedicated to research into socio-economic dimensions of Indian society. Dr. Ajit Ranade is an economist of great eminence and also a political analyst. He brings incisive thoughts on the social, political, and economic factors that are shaping India. His illustrious career has spanned both academic and corporate assignments. He was earlier the Group Executive President and Chief Economist with the Aditya Birla Group, an Indian multinational $60 billion conglomerate with presence in 36 countries. Previous to that, he was also the chief economist at the ABN AMRO Bank and has served as a member of board on several companies and trusts. Dr. Ranade is a member of the executive committees of several Apex industry chambers. He is a member of the National Executive Committee of FICCI and the Economic Policy Council of CII. He chairs the research advisory panel of the Indian Institute of Banking and Finance. Dr. Ajit Ranade is also the Government of India's nominee for the governing body of Economic Research Institutes for Asian and East Asia. He has also served on several committees of the RBI, including the Committee on Fuller Capital Account Convertibility and Committee to Review FEMA for Individuals. He chairs Research Advisory Panel of the Indian Institute of Banking and Finance. Dr. Ranade also writes regularly in the business press on economics and current topics, and you will find his columns regularly in First Post, Business Standard, IBN Live, and the Economic Times. He has authored several books, and most recently he has co-authored an award-winning book titled Rising to China Challenge. A few of his other books include Life Insurance in India, Emerging Issues, and so on. Dr. Ranade is the co-founder and trustee of Association for Democratic Reforms, which is a citizen watchdog body. He's also the trustee of Pune International Center and an honorary senior fellow at the Takshashila Institution. Academically, Dr. Ranade holds a BTech in electrical engineering from IIT Bombay. He received his PhD in economics from the Brown University in the US, and he is also an alumnus of the IIM Ahmedabad. He is a recipient of many prestigious awards. He received the Distinguished Alumnus Award from IIT Bombay in 2009, the Scholar of the Year Award from Wadia Trust in 2010, and the Dr. Ramchandra Parnekar Economics Award in 2012, and finally the Aditya Birla Leaders Award in 2015. Sir, we are delighted to have you with us today, and we look forward to listening to your lecture. Ladies and gentlemen, please welcome Dr. Ajit Ranade.
Thank you, Dr. Ashish Lele, for the very kind and generous introduction. Good evening, ladies and gentlemen. And I am delighted, honored, privileged to be here. So let me begin with a special thanks to Dr. Ashish Lele and, of course, NCL for inviting me. It's a wonderful feeling to be here among so many friends also. And listening to Dr. Mashelkar, you know, you've had a sumptuous feast. I was going to say first it began with dessert. I came a little bit late, so the dessert was just being wrapped up. And then you had a sumptuous feast. And then, of course, you also had dessert. So I thought my talk is going to follow. So the starters are over, the feast is over, the dessert is over. So my talk is going to be a short coffee. And then you will actually have your tea and coffee break. So I'm just going to dwell on a few things. Dr. Lele told me that it's going to be a popular lecture. So I didn't quite understand how to make it popular. But I thought, okay, this is a free license to generally, you know, share some thoughts. But before I begin, I want to especially congratulate an old friend and fellow, you know, journeyman in our IIT board days, Dr. Shivram. Hearty congratulations on your 75th birthday. And Mrs. Rama, very nice to be here, and thank you. This is a double delight for me to be here on this occasion to felicitate Dr. Shivram. I know that, of course, Mr. Mashelkar is identified with NCL a lot, but I believe the second after the Sachin Tendulkar, there was another batsman who also was at the crease for almost 10 years. So he's as much part of this innings of NCL. And of course now Dr. Ashish is at the crease. So best wishes for a grand journey. And so I'm just going to pick 2 or 3 themes from actually Dr. Mashelkar's talk. So that's the way it is going to be popular. It's a evolving talk. So they say that, you know, not once, but 2 or 3 times I was asked on the way 3 days ago, even while coming to the stage half a minute ago, do you have a presentation? There is a lot of dread and, you know, there's a kind of anxiety in that question. Because if I said yes, the first thing that intelligent scientists look, and if there's a fumbling person at the podium, when they put up the PPT, they sometimes, you know, Dr. Mashelkar doesn't do it, but some of the fumbling scientists, they don't go to full screen immediately. The first thing intelligent scientists do is see at the bottom left corner, how many slides? So I have good news for you. I have no presentation, and therefore 0 slides. So I thought I'll just pick on 2 things. One is just by way of my own little starter appetizer for you. Mr. Mashelkar said, look for anomalies. So, you know, I wear a hat as an economist. Of course, I've been happy having had engineering education and so on. But these days, I primarily wear hat mainly as an economist. And sometimes economists take liberties of pontificating and pronouncing on every subject. Polymer science, call me. or elections, or stock market, cricket. So you have to give us the liberty and indulge us. So I thought, so let me look at it from an economic lens, shall we say. And I do want you to have some takeaways from today's talk, even though you had a very, very wonderful feast. So if you remember, Mr. Mashelkar said, look for anomalies. When you're doing this pole vaulting, when you're doing this innovation research, when you're doing these breakthroughs, look at anomalies, you mentioned. So economics is full of anomalies. Economics is full of anomalies. Let me mention a couple of them. This is an appetizer for you to think about how even in economics, if you have a research mindset, and there are so many puzzles, so many anomalies. Number one, these days, a lot of us, I find scientists are not excluded. Doctors, surely, lawyers, and of course, businessmen. You tend to be glued to CNBC or television, or nowadays on the digital screen. So they say that the stock market crashed. These days there's a lot of apprehension because the Fed is tightening interest rates and monetary tightening is going on, and there's Ukraine war and this and that. So the stock market crashed, they say. It crashed by, actually in last 2-3 days it's doing the opposite in India, but worldwide there's apprehension about stock market crash. And it's actually happening in the United States, in parts of Europe. So they say that the stock market went down by 1,000 points. Stock market has crashed. And then they say there was a wave of selling, selling pressure. I hope do some of you own stocks like shares in this? Nobody? I don't believe that. Yes. You certainly own mutual funds. And if you're not owning, then I'm not a mutual fund salesman, but you at least have an EPFO account, right? The Provident Fund. So those guys are investing in the stock market. So they say there's a wave of selling. So what I want to point out is that in the stock market, if there's a wave of selling, and that's how the stock market crashes, and the headline always, it sort of spooks you by saying there's a wave of selling, everybody's selling stocks. But the way the stock market works is that it's a market where there are buyers and sellers. Not even a single share can be sold unless there is a buyer. For every seller, there is a buyer. So if millions of shares were sold today, it also means that millions of shares were bought today. So why doesn't the headline say there was a wave of buying today? Anomaly number one, just to think about. So this language and narrative, the way the sensationalism, it hides the fact that actually the stock market is supposed to be a place where it is anonymous, order book matching, electronic, and every buyer has a seller, every seller has a buyer, and in fact the order is fulfilled. So you never have to worry that I've bought something, but that what I bought will not be delivered to me. That's why the stock markets exist. But strangely enough, when it's falling, we say it's wave of selling. When it's rising up, we say there's wave of buying. Arey Baba, it's both. For every sale, there's a buyer. Second example, and I'll just keep it brief. This is my favorite example, so I like to quote. Have you heard of Mary Kom? She has been the world boxing champion for 6 times. Now, this is an individual sport. And this is, you compete with all nations, 191 countries. This is not some cricketing, 6 countries away, but let me not get into cricket because I'll get into trouble. And I know Dr. Mashelkar is here. Imagine being the world champion for 6 times. And I think the last 2 times was after she had at least 2 children, 3, after being a mother. And of course, you know what kind of hardships. So they also made a movie about her, biopic. It's called Mary Kom. Have you seen that? So there's a lady called Priyanka Chopra who played that role. You know Priyanka Chopra, right? So the anomaly here is that, it's not an anomaly because I just want to provoke you, is that Priyanka Chopra, of course, got a national award, by the way. Getting a national award is more prestigious than getting the Filmfare Award. Priyanka Chopra got paid for that one movie more than the entire lifetime earnings of Mary Kom as a boxer. So tell me how that works. So this is again anomaly, but I mean I could go on. These are puzzles. Maybe some of you think it's not puzzling because you already know the theory and how it works. You know, everything is about supply and demand. But these are the kind of anomalies that sometimes you kind of think, is the economic system working? I mean, here's somebody who's actually done the hard work of getting the boxing career, and you just play that person in a movie and you make actually more than the... So that's what, why he said that anomalies are important, because they make you think. They are not really anomalies when you go deeper into it, but then you start seeing the underlying pattern. Economics or any research is about pattern matching. You see patterns where other people don't see patterns, and then you construct a theory, underlying theory. Then you see how that is explained. And if you're lucky, or if you make progress, then you actually have a unifier theory. That means you have a theory which generalizes. So that is, that's why anomalies are important. But let me quickly move on to the main thing that I want to talk about, and which also was mentioned by Dr. Mashelkar several times in his presentation, which is about inequality. Actually, we were fortunate to interact with Dr. Richard Roberts, who was here and who gave the Bhatnagar Award to Mr. Kulkarni. So this, he spent some time in Pune city, and then we got a chance to interact with him. And he also pointed out that there is a lot of inequality in the world. I mean, I was very happy and impressed that he was very concerned about it. We started out by saying, he was saying that GM foods, or GM, you mentioned GM brinjal, BT brinjal in your presentation. So he was saying, actually, it can do a lot of wonderful things to meet the, India unfortunately has ranked 107th or something out of 121 countries in the World Hunger Index. There's a lot of debate going on because nowadays WhatsApp University is much faster than NCL or any other university. But nevertheless, it's not something to be proud of. And our own data, our own research shows, the National Family Health Survey shows, I mean, repeatedly, because it's conducted every 5 years, that we have almost 1/3, 34% of children in India, according to our own research and our own surveys, government funded, are 33% wasted, something like 30-40% stunting. Wasting means underweight, that is your weight to age ratio, and stunting means height to weight ratio. So there's very large scale wasting and stunting. And now I also hear that anemia is a big problem, not just for children, but also women. So these are all real concerns. And he was saying that in that interaction with Dr. Richard Roberts, he was saying that, in fact, if you allow GM modified, the gene modified foods, which have done wonders in the West, it will go to a great extent to meet the nutritional needs and so on. And it's very much needed in developing countries like India. But then we were asking him, why doesn't it happen? Why are GM foods so much anathema? After a long time, we've got mustard. But of course, BT cotton, which is again genetically modified, is a great success. And they started using modified cotton, GM cotton, in Gujarat, I believe, sneakily. And suddenly, Vidarbha, which used to be a capital for a cotton growing region for India, found that it was vastly overtaken by Gujarat. Then some people went there and said, what are you guys doing? I mean, there's something. Then they realized that this BT cotton was, just roaring. And then, of course, I don't know what is the current status. I think it's officially now okay. It's got official blessing. But that is just an example of success. And this is not a food. It's not something we consume. But now it's gone to mustard, and maybe, I don't know. I'm not, by the way, just like I'm not a mutual fund or a stock market salesman, I'm not a GM salesman, I'm not here on behalf of any company, but I'm just quoting what Dr. Richard Roberts said, which I think made a lot of sense. He was saying that this will go a long way to meeting your nutritional needs. So therefore, the question we asked him was, why is it not happening? Why is there so much resistance to GM? Why? How many in this people would support extensive use of GM for foods in India? 5%, including the front row. So that just shows, there is small empirical evidence that there is a lot of resistance. and don't please blame Jairam Ramesh for it. I know he was the environment minister. There is somehow a very strong resistance to GM foods. And he was saying, I'm quoting him again, he's saying the anti-GM lobby is very strong, starting with Amnesty, is it? No, starting with Greenpeace. And they're well funded. And so I said, then how come they're not well funded in America? Because America leads the world in use of GM foods. And of course, in Europe, there's much more resistance to GM food, but GM food is okay for cattle there. In fact, all kinds of GM foods are okay. So it's funny because the cattle eats the food and then they eat the cattle. So it's gone into their stomachs anyway, but somehow they don't see that connection. But it is little more, the GM foods widespread use is less than America. But then we asked him, Dr. Richard Roberts, why is it successful in America? And you know, what is the answer for that? The answer, he said, is because the food companies in America, maybe it is ADM, what is that, Archer Daniel Metcalf, is it? Midlands, ADM or Cargill or large food companies, growers, they have a lot of lobbying power. I'm using the word lobbying in a nice way. Okay, so they know the importance of lobbying, and they have got their foot in the door much earlier. So they were able to convince the benefits of GM foods to American public, American lawmakers and influencers. And therefore, the anti-GM lobby is not as successful, or perhaps doesn't have as much deep pockets. So it's a question of lobbying, money lobbying ka takkar. Yeah? So then I said, why not in India? And the obvious answer to that is obvious, because the food companies don't make as much money in India. They have no stakes here, very, very little stakes. So there's no point in trying to lobby and trying to spend money on public education, advertising, awareness building, and therefore, whereas these guys now have learned their tricks, so proactively the Greenpeaces of the world, they're already well established and well funded. So anyway, this talk is not a political talk, but I'm just saying, so this was Dr. Richard Roberts telling us, okay, a Nobel winner in molecular biology. But I think that was a great insight. So then the related question was, or rather not question, my observation was that the same thing happens in the malaria vaccine. You know, they are very close to finding a cure for cancer or something like that. They are very close to finding a cure for Alzheimer's. They're very close to perhaps, I don't know, some decisive progress in the treatment of diabetes or pancreatic cancer. Why not malaria? Actually, malaria and dengue kills a lot of people, not just in India, but in Africa and other developing countries. Same answer, because there's not enough money in it. Why should the rich, deep-pocketed drug companies in the world spend billions of dollars doing research for malaria vaccine? Because the return on that investment is not going to come. I mean, I know some of what I'm saying is complementary or not contradictory, I hope, to what Dr. Mashelkar was saying. He said, be affordable, at scale, a sustainable speed, and but it's not yet happened to malarial vaccine. I know that the malaria vaccine race is now 3 major players. I think Paris, Texas, and perhaps Delhi was involved somewhere. They're going neck to neck. And Mr. Bill Gates has decided to fund a lot of this research. But I don't know where it is. But the basic insight which we had in the GM foods is the same in malarial vaccine, that there is not enough money. Because the payoff for this discovery is from poor people, and they're not going to pay you more than a few dollars for a malarial vaccine. And then how is the drug company going to make profit? So one more example, and then I'll come to the main part, then I'll wrap up. That vaccine, so actually after all this, drug discovery takes $1 billion. Typically the estimate is that for one new drug, new molecule, you typically have to spend $1 billion, one unicorn in the language of Mr. Mashelkar. So $1 billion is a lot of money. And the success rate is perhaps 1 in 10, I don't know. So vaccines, we saw COVID. So if you read New York Times, Financial Times of London, or some of the newspapers, they were estimating we have to, at least it will take 10 years for a COVID vaccine to be developed. And guess what happened? we developed the vaccine in about 18 months, or even the splicing and the synthesis was done, I believe, in just a few months, not even less than a year. So how did the miracle happen? So there, I think what happened is, first of all, science and leading scientists are committed to open source, and I think WHO or some kind of global alliance happened in funding this research, and governments came forward to bulk purchases. So the Americans put, I'm told during the time of President Trump, large amounts of advance purchase was done to these vaccines from Pfizer and a few other companies, even before the vaccine was ready. So the government took a bet. So that is public money. When the government puts in an order for $2 or $3 or $10 billion to Pfizer, not knowing whether the vaccine is going to be ready, that is taxpayer money. The American government was enlightened enough to put large orders. Of course, let's not get into the other controversy about that. The rich countries ended up, hoarding all the vaccines. There was a time when almost everybody who didn't have vaccine hesitancy was vaccinated, double vaccinated, and half of the developing world was still waiting for vaccines. So that is the hoarding phenomenon, but we're not talking about that. But I'm just saying that this was the miracle, that the public funding, this huge amount of funding that's required either to push GM foods or to push the development of the malaria vaccine, or in this case, the COVID vaccine, that big money was put behind it, and it happened. So the question is, which is a question which we ask our undergraduate economic students, should vaccines be free? Or should it be left to people, if you can afford it, you should pay for it? And then naturally people say, well, we get mixed answers. They say that people who can afford it should pay, people who can't afford it should not pay. But then how do you distinguish between the two? Then some politicians say that you can make out from their dress. But I know I do have some shirts which have some holes in them, so I'll show up at the vaccine center wearing a torn shirt. But the idea is that the vaccines should be free because of a simple concept called public goods. That if you get vaccinated, I get vaccinated, then you and I benefit, but 10 other people also benefit, because the chances of the virus spreading decreases. It's a mathematical function. The more the share of the population that gets vaccinated, the less the chance of it spreading. So it's a public good. The benefit is going not only, suppose we make it 500 rupees per vaccine or 700 rupees. In some countries they did charge. Then what will happen is only those people who can afford it will buy the vaccine. So we will have under-penetrated the population, and therefore we run the higher risk of the vaccine and the pandemic spreading, or the pandemic will be delayed that much more. So unless the government makes a push, very big push towards spending money on free vaccines, it will not be curbed in a much shorter time. That is called public goods. Public goods are those where even the person who is paying for it and who's consuming it is not the only person who's getting benefit, but a large number of people are getting benefit indirectly without paying for it. So even unvaccinated people get benefit from vaccinated, because the probability keeps reducing. There are so many public goods around. One example which you may not think of it as public good is primary education. So should education be free? I mean, I know this is a very question which doesn't ring a bell for Indians, because we are used to paying, many people have had education where their tuition fee was 500 rupees or 1,000 rupees or 10,000 rupees. But now the private colleges, Ashoka University, they are charging 15 lakh rupees. Can you believe it? But most people are used to. But this question is relevant in economics, that should primary education be free? Yes, because it has the nature of a public good. If you have educated children, they in turn become part of a more enlightened and educated and creative citizenry, which in turn contributes to better, like Dr. Marshall said, better functioning, robust democracy. Because an uneducated mass is not conducive to a robust democracy. So we have to invest in primary education. When I say primary, it should be, of course, quality education. So that's an example of a primary good, which you normally wouldn't think. Everybody, all of us have benefited from education. I benefited, I went to IIT, my tuition fee was low, but the biggest beneficiary of that education has been me. So is that a public good? I mean, did somebody else get benefit of that? Maybe. But the same logic does not hold true for primary education, because maybe for secondary education and for higher education, more and more benefit goes to the person who's paying for it. I didn't even have to pay much because the tuition fee was low. So actually, my education was subsidized. But it's an ulta logic. It has to be for primary education. So that is the point. So this part I just want to conclude and quickly go ahead. So public goods, funding of public good is important because it benefits the public at large, even those who are not paying for it. So that leads to the main point that I want to make in this, this part of the lecture is about inequality. The reason we have to fund public goods, the reason we have to focus our resources on things like the vaccine or GM food campaign or malarial vaccine, all this requires public money. It's taxpayer money, it's government money. In fact, we have been running a free food distribution program for the last 33 months. 5 kilos of food, wheat or rice or coarse cereal per person, per family, per month for 33 months. 810 million people have got benefited. It started during the pandemic, but it's continuing even to this day. It's been extended to December and maybe it'll go further. Some people think there's going to be some electoral advantage to it, but that's what this freebie debate and all that was there. But basically, this is an example of another public good and it's directed towards nutrition. So I want to just dwell on this one question. So these are the 4 or 5 questions I just want to quickly dwell on. Number 1, is inequality increasing? Okay. Number 2, is that bad? Is inequality bad? Number 3, how much inequality is bad? If it is bad, shall we go for zero inequality or how much of it is bad? And number 4, what can we do about it? So the 4 questions I want to and then conclude. Number 1 is inequality really increasing? Number 2, is it bad? Is it a bad thing or is it just inevitable? Sometimes there's pollution. The Delhi people they're suffering from AQI. So it's bad, but what can? So is it really bad? Third question is, how much inequality should we then have if you want to reduce it? And then fourth question is, what can we do about it? So these are the 4 questions I want to, so these are somewhat related to what Dr. Marshall also said. It's related to you scientists also. Number 1, is inequality increasing? Yes, right? I hope nobody in this audience will disagree with this hypothesis that inequality in the world, in India, in Maharashtra, in Mumbai, in Pune is increasing. Now, inequality is measured in many dimensions. It could be inequality just in terms of the income you get. Did you know that if you are making more than about 30,000 rupees a month, I won't ask you for a show of hands, but if you are making 30,000 rupees per month, you are in the top 1 or 2% in India. 98% people are below you. Think about it. 1%, top 1% of India makes 22% of GDP, national income. National income is the income that goes to each man, woman and child per year. That is roughly 250 trillion rupees. Trillion is 10 raised to 9, 10 raised to 12. 210 into 10 raised to 12 rupees is the total value of rupee value of the goods and services that we produce and sell during 1 year. That gets distributed. So the value of the cars and the steel and the telecom services and toothpaste and petrol and diesel, all of it, if you value the production, it is 210 or how much did I say, 250. But that is also income. When a Tata Motors company says our turnover is whatever, 40,000 crores, that 40,000 crores actually shows up as income to the engineers, as payment to the vendors, as dividends to the shareholders, as some money that goes to the Tata Trust. So what is the value of production is also the value of what people are getting. That's why it's called national income. The gross domestic production, GDP, is also national income. It's the same thing. It's just it's an equation, identity. It's just 2 sides of the same coin. But I'm saying if I go to each one of you and say, hey, Mr. Lele, how much money did you make last year? Mr. Mashalkar, I'm not going to ask you, but anyway. And then I do a statistical chart. You know the distribution chart? You'll be shocked to know that it is highly skewed. 1% of the population of India is making 22% of the income. Top 10% are making 50% of the income. The mean income, so total national income divided by the population is called mean income, average income or per capita income. That's about 250 trillion divided by 1.3 billion. Please tell me, calculator. Something like 1.3 lakh rupees, right? 1.4 lakh. So the income per man, woman and child in India is 1.4 lakh rupees. For the whole year, 12 months, divide by 12. 1.4 lakhs divided by 12, 10,000 rupees or 12,000. That is the average. I told you the top 2%, top 1%. So when I say if you're making 30 or 40,000 rupees a month, you're way above the average already. And if the distribution is so skewed, you're actually in the top 2 or 3%. Okay? So inequality can be measured in terms of income. But now you think about wealth. So this is income which is produced every month, every year. But rich people, what do? You can only eat 2 meals of 3 or 4 meals a day, right? You can only wear 1 shirt and 1 pant, and you can only go to so many holidays to 5-star hotels and so on. So they have a large amount of savings. What do they do with the savings? That savings goes into the same stock market, in real estate, in gold, in perhaps foreign exchange, in land. So that becomes their wealth. So accumulated savings from your income becomes wealth. So I can now measure inequality in terms of wealth, not your annual income, but how much wealth do you own? Some of the young people perhaps have not accumulated wealth in your own name, but I know a lot of people who own 4 or 5 flats and a lot of land, a lot of shares in companies. So the inequality is in income, much worse inequality in wealth. Inequality in access. One of the themes you had is accessibility. Access to good education. You have 13 million kids who appear for IIT entrance exam. And number of seats is about 20,000. You go to some parts of eastern Maharashtra or even 100 kilometers from Pune, half the engineering seats are empty. In fact, some schools are offering a scooty. Please come and take admission, we'll give you a free scooter. I think he'll go to a motorcycle soon. So, not just Maharashtra, Tamil Nadu, Karnataka, Andhra, 50-60% of the engineering seats are going empty. And 13 million people are scrambling for this 20,000, 30,000. So inequality in income, inequality in wealth, much worse inequality in access, not just to good education, good health care. And then you talk about digital inequality. 270 million kids, something like a large number, were denied 500 days of schooling during COVID. Why? Because they didn't have a smartphone. People who are doing work from home or classes from home, you only had typically 1 phone per family in poor families. And that same phone had to be shared between 3 kids, but 1 kid is doing SSC exam, uska 10th SSC hai, usko zyada lagega. But then they fight each other, then the phone drops and the screen is cracked, so no phone. And then when they had a smartphone, there was no good Wi-Fi. When they had good Wi-Fi, there was no electricity. So inequality of access. Then inequality is also widening regionally. Some parts of India, you go there, it looks like Singapore. Some parts of India, they look like Africa or maybe sub-Saharan Africa. So there is income inequality, there's wealth inequality, there's access inequality, there is regional inequality, there is gender inequality, men and women. My point is so many dimensions. So my first question was, is inequality increasing? Answer is yes. And it is increasing and increasing and increasing and increasing. It's not going down. In fact, Dr. Richard Roberts also said that he's worried that the US system is wonderful because it's pushing all this lobbying for GM foods, but he said capitalism and market economy is not working because inequality in the US also is worsening. It's worsening in Brazil, it's worsening in Europe. There's 20-25% youth unemployment. So that's my first point. I hope that's established. Inequality is increasing in many dimensions. Number 2, is that bad? Is inequality bad? So would you want to increase your income tax rate to 75% and take your money and give it to the poor? Hey, don't get carried away, he says. So inequality is bad because beyond a certain point, it will create social tensions. social instability. Imagine if inequality becomes so much that people don't have, some people are so poor, some people, incidentally, when I say inequality, when you have economic growth, India may be growing at 8%. So national income, which is 250 trillion rupees, is growing at 8%. But there are some sections of that economy, Indian economy, which are growing at 30%. Some people's income is growing at 20, 30%. Therefore, some other people's income is growing at 0 or 1%. So everybody's income is increasing. I'm willing to accept that. That's not even true, but let's accept that. But the way it is increasing is very unequal, and therefore the gap is widening. Now what happens is, if the income inequality keeps increasing, at some point you're going to have social instability. So all that economic growth that you're seeing because of investment and consumption is going to reverse. So if you're worried, so you should be worried about inequality for nothing else, but at least for your own future and your children's future, because if you don't want to live in a polarized society, and you can have polarization not only in terms of income and wealth, but also in terms of WhatsApps. You know, every family has now 2 camps, either you're pro or against. But so increasing instability, increasing social instability, increasing polarization is bad for the investment climate of the economy, for social stability. And that will deter investment, and that will lead to actually eventually stagnation, if not reverse. So inequality is bad for that reason. I hope you're okay with that reasoning. But I think this is an instrumental view. Inequality is bad because instinctively we don't like it. We don't like to live in unequal societies. You know, we don't like to be in societies where you have to be completely gated. You're sending your child to school, you need 2 bodyguards. Pata nahi kaun snatch karega. You're going, your young daughter is going to work. Be careful, which raste mein. So do you want to live in a society where the law and order situation is like that? But somehow highly unequal. You know, there was a time when South Africa, Pretoria or Cape Town they used to say that after 5 p.m. please don't go outside. You're going for a conference, oh fine, but after 5 p.m. please don't go outside because it's unsafe. Why? Because there's a large black ghetto, which is very poor neighborhood, and then there's these 5-star hotels or whatever, conference venues, unequal societies. So first question was, is inequality increasing? Yes. Number 2, is it bad? For no other reason, at least instability is bad for economic growth. But I think there's a more fundamental reason that human beings are, I think, fundamentally empathetic. So inequality should be, you should have objections to inequality for its own sake. If you have a fellow feeling for your fellow human beings, if you have empathy, if you have some feelings of, then you must be bothered about inequality when you see. That's a little bit of it's not just a moral argument. Neurological, since you guys are biologists or molecular biologists and all, there is now work in, I think, Berkeley, it's already 10 years old, where they have discovered neurons, sensory neurons in the cortex, which get fired. So, if you touch me here, then something in the cortex, a neuron gets fired, and there's a sensory feeling. They have seen that now if you touch somebody else, and I have a phantom arm, I lost my arm in the accident, and then you do something there, I start getting feeling. So that is the neurobiologically observed, confirmed. So I think their empathy is hardwired into our DNA. So inequality is bad not only because of the instrumental view that it will lead to, but it's also bad because we are fundamentally human beings and empathetic. Third question was, so first there's consensus inequality is bad. Second is it is increasing. Second is it's bad. So how much inequality should we accept? Shall we have perfect equality? No way. We can never have that. Shall we have perfect inequality? No way. So what is that midpoint? If one extreme is 0, the other extreme is 1, where do we go? You see, what happens is India is a young country and growing at 8%. That means there are a lot of talented people, like in this room, who are doing innovation, who are doing new discoveries, who are scientists, who are doing unicorns. So obviously, talented people are going to go far ahead. They're going to be millionaires, billionaires, and they'll be far ahead. The gap will widen. So inequality is always going to be there. And we can't curb these people. But how much inequality should we accept? Does anybody have a mathematical answer for that? No. Because Americans, if they do when they take a poll among Americans, the poor people, they're not jealous or envious of rich people. They actually look up, they say, by hard work, I'm going to get there. In Europe, among Europeans, it's thoda zyada hai. They have more antipathy towards rich, poor, some feeling is there. I'm not so sure about India, but I should find out. But it's not as if poor people hate the rich people. Sometimes it's aspirational. So you want rich people around so that they become role models because wealth creators, let me not call them rich people, wealth creators. But how much gap is okay? Is it okay for 1% of the population to have 30% of the national income? They capture this in a statistical ratio called Gini coefficient. Now, how much is okay or not okay is not for you or me to answer. It's not a mathematical formula. It is a sociological, socio-political process. And we determine that optimality through a wonderful thing that India is famous for. It's called elections. So we vote on these issues and we select or vote for political parties who stand for certain things. So this collectively society has to determine how much inequality is okay and how much is not okay. So if you look around the world, the Scandinavian countries like Finland, Denmark, Sweden have the least amount of inequality in the world, Gini coefficient. America is large, but I think India is also large inequality. And Brazil is large. China is a little bit like India. Japan is low, Korea is low, Thailand is low. So every society has to determine how much inequality is okay. But you all have to think about it because we all contribute to it. So that's the fourth question. How much is okay? I don't have an answer. But I feel that 0.9 Gini is terrible. 0.4 is sort of okay, but not really okay. I would be more comfortable with 0.3. But I'm not going to be able to engineer. Even Prime Minister cannot engineer it. This is a collective sociological process. So fourth question, incidentally, when I said inequality is bad, it is like air pollution. When I said it hurts our social tensions and all that, it affects everybody. It's like pollution. So we need to reduce pollution. So we can't reduce it to zero, but we need to reduce pollution. So inequality is like pollution. Think of it like pollution. So then now last question is, what do we do about it? That also is a very big open-ended question. I think the one thing that you should do, and that is where we come back to Dr. Mashelkar's lecture, is that we have to pursue excellence. Actually, all the answers to that last question are there in his presentation because we have to pursue excellence, we have to create value, we have to pursue affordable excellence. So we have to, because excellence in innovation, in science, in engineering, in business or industry creates value. And then the question of reducing inequality is a question of redistributing that wealth. And that is left to our sociological process. There was a time, by the way, when dividend income in India was tax-free. I know that a lot of people didn't raise their hands when I said stock market. I hope you know what is dividend income. Sabko malum hai na? So if I own shares and Mr. Narayan Murthy used to say, or I don't know, somebody else, they say, I take only 1 rupee salary. Arey? But you didn't tell us that you get 5,000 crore income annually as dividend. So, that was revealed, by the way, in just last week because Mrs. Sunak, who's the Madam Prime Minister, they just found out that she gets 200 crore rupees annually as tax-free dividend income because she owns 1% of Infosys or 1.5% or 0.7%, something like that. Oh, so just owning 1% of the shares gets you 200 crore income. What do you have to do for that? Just relax. Because it comes. So that dividend income used to be tax-free. So it used to bother me that somebody like, I'm going to take names, Mr. Nilekani, Mr. Murthy, or say Mr. Ambani, just by ownership of shares, they're getting thousands of crores of income free. When even if you're getting a salary of 3 lakh rupees or 4 lakh rupees, you have TDS. In fact, you don't even get your full. Have you seen how many people get a salary slip in this hall? I hope a lot of people, yes. So you don't even see the full salary, right? When you see it, then pay slip aa gaya. Oh, usme to already cut ho ke aaya. Before you can use it, the government has already got a hand in your pocket. When you are sleeping, they put your hand in. But it doesn't happen to dividend income. So I'm coming to what can we do about inequality. So this is an important political or socio-political or economic issue. Should dividend income be taxed or not? Now, you'll be surprised to know that it took us 20, 30 years for this campaign. But finally, it has happened. It took a long time. But this is the role for you in public debate. You may agree or disagree. You present your viewpoint, I'll present my viewpoint, Mr. Shivram will present his viewpoint, but we need to debate these things. Should it not be the case that you are rich no matter what your income may come as dividend income, your income may come as your patents on your innovation, or it could come from some consultancy you get, or copyright. But agriculture, I know the favorite beating is, favorite thing to beat upon is, hey, these farmers, they're all enjoying and driving their Mercedes cars. Sir, 80-90% of India's poverty is in rural areas among farmers. So there are 140 million small parcels of land in India. So almost all farmers are poor. So when we talk about those poor, farming income should be taxed. There's nobody. In fact, the people who are quoting this so-called tax-free income in farming are actually not farmers. Some of them you can find them in, or many of them in parliament. But anyway. So this is the question to ask. What do we do about inequality? You need to find efficient, fair, just ways of redistributing this. So don't penalize wealth creation. Don't penalize the winners. Don't penalize the innovators. Don't penalize those fantastic innovations that are going on in places like NCL. But you have to agree as a society on what is the best way to redistribute that. So we have decided that one way to distribute it is through free food for 810 million people. I'm not sure that's a good idea, by the way, but it's been going on for 33 months. But can we fund free primary education, high quality? I'm going to conclude with that, but let me just wait a minute. So we should, if you can't give, actually, they are going to talk about UBI, Universal Basic Income. We are already talking about whether we should give income to everybody, free, basic negative income tax to everybody. But you can think of many other ways. Can we fund public goods? Like, for example, you were talking about public-private partnerships. So funding of research, this is a public good. So we should find when there's good research, it creates opportunities for both rich and poor people. So there's an indirect way of redistributing your money, indirectly. When you build a very high, when you spend money on improving air quality of Pune or Delhi, you're actually helping the poor people, because 50% of the population of Delhi is also poor. So air quality improvement is not only for the rich people, but that has to come through public funding. That means it's coming through tax money. So there are many creative ways of doing it. And that's, I think, but that only one person cannot decide. That is a collective, again, process. All of us have to participate in that. I mean, that's the role. You can't just be a scientist in a lab. You have to participate in this public discourse. So I want to just end by saying this, on this note of public, what is the true test, litmus test of that we are, as a society, we need to be, proud of. I won't say developed country is, when we will, of course, those kind of targets are good. We should have at least 10 Nobel Prize winners or 20 Olympic individual sports winners or so many patents. But I think three things. Let me make sure I get them right. So number one is, when you go to a European hotel, it's late night, you just got the train, you landed, took the train, then go to the hotel, then it's late, you're tired, you go to the room, and you just want to drink water, and then you see, where is that? I don't see any water. So you call the lobby downstairs. Hello, yes, madam, can you please send some drinking water? What do you mean? There's water in your room. No, I don't see any bottle. No, from the basin, wash basin. What? So that's number one. When you can have drinking, when you just open the tap and have drinking water from your tap. Believe it or not, this used to be in India as well, at least in some parts of Pune, Mumbai. So first indicator of a developed country is when everybody has very reliable drinking water from as ordinary a source as a tap in your house. Second is when it's not when poor people have cars or scooters, but when rich people travel by public transport. That's an easy one to accept, right? And the third one, which the young parents here will appreciate, when your most preferred school for your child is your nearest municipal school, walking distance. This happens in... Thank you. This happens in Korea, in Germany, in Japan, in any other, in France. So even in America, people actually choose to locate in places like Cupertino or something because the city school, the municipal school is the best. So these three indicators is what we need to strive for, and I hope you got some sense out of this. Thank you very much for your patience. Thank you.
Again, on behalf of our organizers and all of you, we'd love to thank Dr. Ajit Ranade for an absolutely stunning lecture. Thank you so much. While he was climbing on the stage, he told me he'll take 15 minutes. And the last 45 minutes have been one of the most enthralling times in this auditorium. Thank you so much for both of you. Absolutely stunning lectures. You have kept us completely enthralled. I'm going to leave the floor to the conference organizers for the next part of this program.